engineering to advanced level. And on this particular session of this workshop, we will try to learn regarding the current understanding of stem cells with a specific focus on their characterization, function, and role in regenerative medicine. And the course will discuss in detail various bioengineering approaches to stem cell research, including an overview of adult and pluripotent stem cells and their basic characteristics, isolation, expansion, and therapeutic potential of multipotent adult progenitor cells from the bone marrow stromal tissue, and three-dimensional cultural systems, biomaterials, and different biophysical tools used in stem cells research and advancements in the field of stem cells and regenerative medicines. So on today in this session, we have distinguished speakers from National Center for Cell Sciences, Dr. Nivedita Lenka. She is working as scientist F in stem cell biology there. As our next speaker, we have Dr. Parul Johri from MIT Institute of Biotechnology, Lucknow. And the third lecture will be delivered by Dr. Deepa Subramaniam. He is working as a scientist in stem cell biology, National Center for Cell Sciences, Pune. For this whole program will be coordinated by Dr. Sugandha Singh Madam. She is working as Head Department of Biotechnology in Sant Hirdaram College, Girls College in Bhopal. She has accepted our invitation to coordinate this particular workshop. And I also welcome as a convener of this particular workshop, Dr. Nivedita Lenka. Ma'am, so that she will convene this whole workshop as a convener and will enlighten and also will provide concluding remarks on this particular session on stem cell engineering. So firstly, I would like to invite Dr. Sugandha Singh ma'am to coordinate this program, and then first lecture will be delivered by Dr. Nivedita Lenka ma'am. So Dr. Sugandha Singh ma'am, please.
Yeah, sir. Thank you. Thank you so much. So a warm welcome to all the honorable speakers in today's workshop. I heartily welcome all the recipients, the members of academic fraternity and students who are a part of this workshop today on stem cell bioengineering. This is a very exhausted, voluminous program which is being organized by the Department of Biotechnology, Faculty of Life Sciences and Technology, AKS University, Satna. Today's workshop on stem cell bioengineering, and this series of workshops, this has been started since long, from September 2020 onwards, and this is likely to end on 21st of October. And I again extend this opportunity my thanks to Professor Kamlesh Chaure sir, under whose able guidance and stewardship this series of long voluminous workshops is being organized. So once again, my heartfelt thanks to you, sir, for giving me this opportunity also to be a part of this academic gathering. So friends, this stem cell bioengineering workshop is going to be a very important lucid instrument, which is going to play a very important role also in addressing the issues like cure of diseases by providing treatments, whether it is not many researches have been going on in this field, whether it is Parkinson's or Alzheimer's or any spinal cord injuries. Stem cells have been a major tool in addressing all these issues, and stem cell therapy has drawn considerable attention, since the transplantation of stem cells can overcome the limitations of autologous transplantation of patients' tissues. So we are going to get the insights and expertise of our honorable speakers in this long day workshop. And before inviting our first eminent speaker, Dr. Nivedita, I would like to throw some light on her achievements and introduce you all to her. So if I talk about her education, she is PhD genetics from Osmania University, Hyderabad, India. And she's been a postdoctoral research associate from Temple University, Philadelphia, USA. She's been a postdoctoral fellow, again from University of Pennsylvania, Philadelphia, US. And in 2001, she was a postdoctoral research scientist from University of Cologne, Germany. And presently, she is associated with National Center for Cell Sciences, NCCS, Pune, since 2001 and working till date. So she has to her credit many publications, where her recent publications are like augmenting the differentiation potential of mesenchymal stem cells, which was published in Cytotherapy. Then influence of doping ion capping agent and pH on the fluorescence properties of zinc sulfide quantum dots, so on, and many more such publications. She has given her expert and able guidance to many of her students, and she's been credited for a patent also, that is a new molecule for cardiac development promoting activity that was granted in 2005. The work and the research which she's been doing on has been supported by NCCS intramural funding and various project grants received from Department of Biotechnology, DBT, India. She has memberships of many professional bodies like American Society of Microbiology, Stem Cell Research Forum of India. She's a life member of this body, International Society for Stem Cell Research. She's been continuing as a member. Then she's a life member of Indian Academy of Neuroscience. Then she was a member of New York Academy of Sciences from 1998 to 1999. She's been a recipient of many awards and honors and recognitions. She's been the member of Editorial Advisory Board in Annals of Neurosciences 2009. She's been a visiting scientist and JSPS DST Exploratory Exchange Program under the Indo-Japan Cooperative Program between Japan Society for Promotion of Sciences, that is JSPS, Japan, and DST India in 2007. She's been a member as an expert in Faculty Selection Committee in All India Institute of Medical Sciences, New Delhi in 2005. Then she's a member in Scientific Program Committee and 22nd Annual Conference of Indian Academy of Neurosciences, that is in 2004 and 5. She served as a member of subcommittee that was formulated by DBT for the site visit to CMC Vellore in regard to monitoring the project which was submitted to DBT in 2004. And she's also been the recipient of many travel awards, and a few of them to include International Society for Stem Cell Research Travel Award in 2008. And likewise in 2005 also, she's been the recipient of Rockefeller Foundation Award in 1991. She's been the reviewer of many scientific journals like Current Science, Stem Cells, Journal of Tissue Engineering and Regenerative Medicine, Aquaculture Research, Indian Journal of Experimental Biology, and has been reviewed many research projects for funding agencies also. She's been the examiner also for evaluating the PhD thesis submitted in genetics and biochemistry, also in applied biology. So we are very fortunate today, ma'am, that you are there amongst us, and you will be throwing light on the same burning issue and the possible interventions making use of stem cells and dealing with all sorts of problems and finding a cure for the same. So we happily invite you. We are delighted to invite you for your lecture, ma'am. So over to you, Dr. Nivedita, ma'am.
Ma'am, your screen is visible. Yeah. Please minimize it, ma'am. This screen. Okay. Please hide this bar, ma'am. Click on hide. Yeah, that's fine. Thank you.
Hello, am I audible?
Yes,
Yes,
ma'am.
yes.
Absolutely.
Okay. No, that's what I was not understanding whether I'm audible or not. Okay. So, as I told, thanks a lot, Dr. Sugandha and Dr. Chauri also for the kind invitation. So, I'll start giving some introduction on stem cells and what are the potential implications. So, the title that I have chosen for this morning's topic is STEM concerning stem cells. As all of us know what is STEM, that is S stands for science, technology, that is T, E engineering and M mathematics. So you must be wondering what is STEM concerning stem cells. So nowadays people are talking about the interdisciplinary science. So the stem cell is another arena or the avenue where actually you are incorporating the knowledge from all the fields to address the challenges that this field is actually experiencing, as well as how to exploit or find out the potential implications in a much better way, utilizing the knowledge gained from all the fields. So I will also walk you through how this knowledge from different fields are brought about in understanding stem cells and the potential implications. So basically, since this is attended by many students and researchers, I would like to point out in the beginning. So when you are talking about science technology, so what basically science is? Is it just talking about science per se is good enough? No, it is actually exploring the possibilities. How will you explore the possibilities? By asking questions like what, why and how. And that is what most of the researchers will always do. And this science or scientific innovation is not just limited to different areas like physics, chemistry, maths or biology. It's basically intercalating the information that is gained from all the field and bringing about the change and asking the questions, as I mentioned, what, why and how. So that is what we have to also understand. What are the stem cells? How will you get and what are the implications of stem cells? So as rightly French physiologist Claude Bernard in 19th century he had told, the joy of discovery is certainly the liveliest that the mind of human can ever feel. And those who don't know the torment of unknown cannot have the joy of discovery. So with these few words, I just go to stem cells per se. So what are questions one can ask and how to address all those questions or the curiosity of the mind that we are all having. So we have to actually inculcate the curiosity of a child. As we know, the child always asks a lot of questions. Many a times parents get worried what sort of question the kid is asking, so we have to be well educated to answer that. The same the generation of the students or the researchers, they also need to be more curious like when they were kids. And the question answer, when the mentor asks you the questions or you are asking the question to mentor, it should be much more like a mutually agreeable, like not agreeable only, arguable also. So you ask questions and try to address those questions, the answer to those questions. So this way actually the research can go forward and we can address many more aspects of any field that we are working on. So today's topic is on stem cells. So I will walk you through how this field is important, how it has come and what are the potential implications. So as I told, the questions that you have to ask is what, why and how. So this is how I have mentioned actually in my slide. So STEM, the first two are science and technology. So there we have to ask what are the science or what is the science involved in and what are the different technologies involved to carry out this work. So what are the stem cells? Why you have to work on stem cells or why you need to understand stem cells? Then the question comes, how would one study stem cells? So what are the tools, technologies, or the strategies that you would like to use for understanding the stem cells per se, as well as exploring this field further, what is totalitarian utility and what is the aspect of engineering mathematics you are using? So this aspect covers how do you engineer different organs and in a minuscule level the organoids, even bioimplants, using the AI or mathematical modeling. So as you know, all the fields like STEM truly represents. So in understanding how the stem cells function and how to understand the stem cells per se. So the first question is, what are the stem cells? So stem, as the title says, STEM and stem, those are two different aspects. So stem in literally sense, that is the, like it's the branch or the origin. So you think about a building. When a building is constructed, so you are putting a foundation stone. So similarly, when we ourselves have to imagine, you, me, everyone, so how we all have come into being. So this is from a single cell, the mother or the founder cell. So stem is the branching or the origin. Those are actually serving as the cellular architect, which are putting the building block from which single cell, those are formed under tissue. Further in the hierarchy, the organ is developed. Then the entire organism like you and me were coming. So this itself indicates that the stem cells, they are residing at the bottom or the founder members. So those who are actually giving the shape, entire blueprint of the organism, the cellular blueprint, that is what is ingrained within this cell and you are getting the entire organism. So if you know these are the stem cells, then the questions come, is it only the single cells from where we all have come into being? That is the only cell which is stem cell or there are different stem cells? So what are the different types of stem cells? So in that case, what are the different sources? Then what is the mode of isolation if you isolate? How to characterize? Then how to culture, expand? And can you differentiate and get the cells of your interest? Can you enrich, purify? And if you do all these things, will those be physiologically active? Then finally it comes, everything is addressed. Finally it comes, what are the typical implications? So to address these questions, the same thing, how and why. So in this case, as I told, we all have come from a single cell, that is zygote. So if you can see here, from the zygote, once the zygote divides, then that is zygote is having the cellular groupment, so we are getting the entire organism. So in total, it is giving the entire organism. The next slide, I will talk about the differentiation potency. There you know the zygote or 2 to 4 cell stage. Since those are capable of giving rise to the entire organism, those are called totipotent stem cells. From that, you can get the totipotent stem cells. And then further, when the zygote divides, it gives rise to 2-cell, 4-cell and followed by the morula, that is the first developmental stage, which is 8 to 16 cell stage. Followed by you are getting the blastocyst, where the outermost layer, that is the trophoblast or trophoectoderm, and then you have got a cavity blastocele, and this is the inner cell mass. I hope you guys can see the cursor moving. So this is the inner cell mass or ICM in short. From these cells are the ones which can give rise to the embryo proper. That means the entire embryo development or all the organizational blueprint is there within these cells. And what is the role of then trophoblast, the outermost covering? That is giving rise to the extra embryonic part. So that is what providing the trophic support. The placenta, that is what is coming from the trophoectoderm trophoblast, and that supports the ICM derived embryo proper. Then further, so these ICM cells, since these are giving rise to the entire embryo, so from that the stem cells are isolated. So that is at the embryonic stage before implantation, and these are the cells, when you isolate the stem cells, those are called embryonic stem cells. In human, this actually commences around 5 to 7 days. In rat or mouse, it's 3 to 5 days. So further during development, these blastocysts undergo gastrulation. So during gastrulation, you are getting the 3 germ layers: ectoderm, mesoderm, and endoderm. As you can see, the mesoderm, from the mesoderm, you can get all the connective tissues, muscle cells, hematopoietic, those are the blood cells, mesenchymal cells. Same from ectoderm, you can get neural cells, skin cells. And the endoderm derivatives are long pancreatic cells, those are the ones. So during gastrulation, the cell plate is determined to 3 germ layers and the derivatives of 3 germ layers. Further during development, you are getting actually the fetus is coming. In fetus itself, there are different cells, and these also give rise to germ cells, which are also having the same potential like the ICM derived cells. So the germ cells, PG or EG cells, those are called primordial germ cells or embryonic germ cells. They also contain the potential of giving rise to all the cell types except the extra embryonic one, which are coming from the trophoblast layer. That's why these are called pluripotent stem cells. Then if you are getting any of the stem cells from the fetal tissues, those are called fetal stem cells, and if you are getting from the adult, those are called the adult stem cells. So the next slide, actually I have categorized these stem cells as you could see from the schematic one previously I showed. So the types of stem cells, it can be broadly categorized into 2 types: embryonic and the second one fetal or adult, depending on the source of derivation. The third one, which has been mentioned here as cancer stem cells. So the recent dogma states cancer recently, because we know it's a dreaded disease. The basis of cancer also now it has come down to the stem cells are the whatever is there in our body, the adult stem cells. Those are the ones which are probably giving rise to cancer, and those are coined as cancer stem cells. So what is the basic difference between cancer stem cells and normal stem cells? They, in fact, if you compare the embryonic stem cells and cancer stem cells, there are many commonality. Many genes are expressed commonly in both the cell types. Then embryonic stem cells, if you compare, since it is giving rise to the entire organism, or it is capable of giving rise to all the cell types, because those are derived from the ICM, which is giving rise to the embryo proper. So the development proceeds in a very, very organized fashion. Everything is controlled, whereas in cancer, it is uncontrolled. So when the adult stem cells which are residing in our body in a quiescent state, due to some insult, they get back to the mitotic state and they go haywire. That time, that is the origin of cancer. So those cells which are residing there and which are giving rise to the cancerous tissues, the stem cell population are called as cancer stem cells. So these are based on the sources we know, or based on the differences of potential, as I mentioned earlier in the earlier slide. So we can discriminate those into totipotent, pluripotent, multipotent. There is another one also, oligopotent and unipotent. So totipotent in toto, as I mentioned, the zygote and 2 to 4 cell embryo. So those are the ones, because that time it is prior to the implantation, and those ingrain the capacity of giving rise to embryonic and extra-embryonic part. That's why they are actually capable of giving rise to the entire organism. That's the reason why those are called totipotent. Next in the hierarchy, the cells are called pluripotent. So the pluripotent cells, those are derived from inner cell mass of cells in the developing embryo, which are giving rise to the only embryo proper, the embryonic part, not the extra-embryonic part. That's why those are called pluripotent. Then further down the line, those are multipotent, which are tissue restricted, or unipotent, those are also tissue restricted, those are found in the adult body. So in our body itself, in different organs, we have distinct population which are called stem cells, and those remain mostly quiescent. As and when there is any injury, they participate in the cell cycle, they divide and differentiate. So at the bottom you can see I have listed some of the sources which are actually coined as a tissue restricted fetal or adult stem cells. The best being, as you know, nowadays many people, there are a lot of advertisement earlier also to store the umbilical cord blood, saying that it may help in the future the stem cell therapy. So basically umbilical cord blood, amniotic fluid, placenta, those are the waste product during childbirth. But you can also isolate stem cells from that and further implications can be achieved using those. So this is basically waste. It's a kind of you are utilizing the waste product for gaining something. So these are the different kinds of stem cells or the types of stem cells that one can probably study, and depending on the interest that you are interested in, what you want to study. So the same question comes, what you want to do, then question comes how you want to do. So now we know the categories of stem cells, then further the question comes, okay, if you know the sources, then how will you isolate or get the stem cells in hand for further implications. So first thing is embryonic stem cells, as I told, and okay, I just before I forget, here in the pluripotent cells I have mentioned about, one is inherent type, one is reprogrammed. So what is inherent? So which are coming from the developing embryo, those are the actually inherent pluripotent stem cells. And the categorization under these comes under embryonic stem cells, as I told, from the ICM derived, then primordial germ cells or EG or PG cells, even the embryonal carcinoma cells, which can give rise to all the three germ-like derivatives, ectoderm, mesoderm and endoderm. Those are the inherent pluripotent stem cells. Then what is reprogrammed? Reprogrammed, that is rewiring. So you are introducing pluripotency in some cells, those are basically somatic cells, or maybe adult stem cells. So you are bringing back to the pluripotent stem cell stage. So that's why those are called reprogrammed cells. So for these inherent cells, embryonic stem cells, the Nobel Prize has gone to Martin Evans and Kaufman. So they are the pioneer in isolating the embryonic stem cells from the inner cell mass, and the reprogramming or the reprogrammed cells, which are called the induced pluripotent stem cells. In 2012, the Nobel Prize has gone to Shinya Yamanaka from Japan. And at the same time, another way of reprogramming, that is called SCMT, John Gordon, who was the pioneer. So both of them have shared the Nobel Prize. So as you can see, this field, it's not, neither it is recent, actually it dates back to 1960s or so, when the adult stem cells were already known, but not the embryonic one. Embryonic one came in 1980-81. After Martin Evans and Kaufman, they found this embryonic stem cell. Okay, going to the next slide. So as I was telling, how will you get the stem cells? For embryonic stem cells, the pluripotent stem cells, so the inherent one you can get from the embryo. So you can do either direct embryo culture. I will also explain what is immunosurgery or laser microdissection. So when you have the embryo, so there I will come in the next slide, I will show you how the immunosurgery is done, or you can also use the laser microdissection. So there what you do, the outermost layer, actually it's the mucilaginous one. So you have to zona pellucida, you dissolve that. In case of immunosurgery, you dissolve using some kind of enzyme that was in the 50 days. After dissolving, you have got the trophoblast layer. So that you have to do, use specific antibody which will add to the surface of the embryo. Then use the complement which will chew off wherever the antibody is bound. Then the inner cell mass exposed, that you can culture further. And under laser microdissection, you just apply laser beam so that you can extract the ICM cells and grow. And embryo culture, directly you can culture the embryo, provide specific factors for their growth, and then you can provide the factors which will support the ICM cell growth, and you can get the embryonic stem cell growth. So these are the inherent ones. Then as I mentioned, you can also use somatic cells, reprogram those. And how will you reprogram? So every cell type has its own identity. So if you are rewiring the system, any somatic cell, you are bringing back to the embryonic stage. So you need to introduce specific factors, specific genes, which will make the cells which are somatic cells or more mature cells to come back to the naive state like the embryonic cells. So that is done by embryonic gene transfer. It was actually pioneered by Shinya Yamanaka's group in 2006-2007. So they have used specific genes, the cocktail of factors which are expressed in embryonic stem cells. From 24 such genes, they have it like cut down into 4 genes, and they have identified those 4 factors which are currently called Yamanaka factors, introducing those genes to somatic cell and making those, bringing back to a naive state like embryonic stem cells, and those are attaining the pluripotent status, and those are called reprogrammed pluripotent stem cells. So now we know these are the sources and how will you isolate these cells. So coming back to somatic cell nuclear transfer of the reprogramming, this is one way. As the name suggests, so this is from the somatic cell, you can transfer the nucleus to another cell. So here, what are the requisites? You require a somatic cell basically. And where will you transfer? So for that, you select X or Y sites. As this particular scheme or the pictorial depiction. So why it is required? Because in embryonic stem cell, if you think it is coming from the developing embryo, so you are taking the inner cell mass cells, so you are destroying the embryo. So that's why it actually invites lot of criticism or ethical concern. So why to destroy the embryo? So there are a lot of discussion, debates actually had gone into this particular field of research, embryonic stem cell research. So the other way, how to actually curb this ethical dilemma. So one way, if you can take the somatic cell, so there you are not destroying any cells. So if you are taking somatic cells, so as I mentioned, the embryonic stem cells, those have got the potential of pluripotency. That means they can give rise to all the 3 different germ layer derivatives. Adult stem cell in the hierarchy, those are tissue restricted. That means they are residing in specific pockets in our body. So what is the meaning of that? So they are more restricted in their potential to divide and differentiate into the cell types. An embryonic stem cell can give rise to all the 200 and odd cell types in our body. Tissue restricted stem cells, for example, if you are taking from neural stem cells, those are there in our brain, CNS and PNS. They can give rise to neuronal cells and glial cells. Glial cells, astrocytes, oligodendrocytes. So those are actually falling under the category of multipotent stem cells. But basically, they are responsible for giving rise to neural cells. Same also if you are taking hematopoietic stem cells, those are giving rise to all the blood cell derivatives. Similarly, any tissue restricted, they are basically capable of giving rise to the cells of that particular organ or tissue. So they are more restricted in their differentiation potency. And same is true also for the renewal, how much they can divide. While embryonic stem cell, they can divide indefinitely. The adult stem cell, they have restricted cell proliferation, the self-renewal characteristics. So in my subsequent slide also I will show you the characteristics features. There you will come across the self-renewal. That means you are giving to the cell self-renewing. So embryonic stem cell, they can renew indefinitely. Adult stem cell, they have got the restricted self-renewal capability. So in that context, they have got some limitations. But when it comes to therapy, if you are taking the stem cells from a patient, so in terms of, because if you are giving to any patient for therapeutic implications, there is, if you are giving to the same patient, it is autologous, so there are no immunological complications. But if you are doing allogenic, taking from another patient, so it is becoming allogenic, then there you have to actually address the immunological complications. So in this context, so if you have to combine the beneficial aspects of the properties of embryonic stem cells which are pluripotent, then adult stem cells, especially the autologous cells, which you have to make patient specific. So in this case, you have to think some way you have to club the potential benefits of both cell types and bring together. So somatic cell nuclear transfer also addresses this. Same also is true for induced pluripotent stem cell. I will talk about that later. So in this, what happens if you want to take the cells from the patient, any cells, and you want to make patient specific cell lines, which you have in mind that you have to implicate further in therapy. So you take any somatic cell and it has got the nucleus which is ingraining the blueprint of the organism in terms of the genetic profile. So at least if you have the same profile, you will not have any immunological complications. So you are taking the egg or oocyte, try to enucleate. Remove the nucleus So it doesn't have any genetic elements here, but it has got the ooplasm. But after that, you take any somatic cell, take that, either you take the nucleus and this enucleated egg, you introduce the nucleus to the enucleated egg, or you try to fuse both. After that, you stimulate the cells to come to the stage, it's by chemical or mechanical means, where it attains the stage of blastocyst. Once it becomes blastocyst, you take the inner cell mass and then grow. Then you are getting the embryonic stem cell from this. So this is at this stage, these cells are called EMT somatic cell nuclear transfer is achieved, and this steps when you take, that is falling under therapeutic cloning. But if these cells are implanted back to a surrogate mother, and from that pups are born, that is called reproductive cloning. That can be done only in the veterinary field to get superior or super herds. But in human case, it is ethically incorrect because it's not permissible area of research, and you cannot get the reproductive cloning done in human. That is why we restrict ourselves to the therapeutic cloning aspect. So we take patient-specific cells, enucleate oocytes, and clump together and get the blastocyst and derive the embryonic stem cells, and then further downstream applications. So these are the steps which are noted here. So this was done in the next slide I can show. As early as 1952, this concept was already there. Further, it was by John Gordon 1962. So here both the groups, they have shown using the frog embryo and the tadpole. So in this case, the first case, they have taken the nuclear material from the frog embryo and taken the oocyte from egg cell of the oocyte, and there they have enucleated and introduced the genetic material, the nuclear material from the donor, that is here from early stage embryo. And what John Gordon has done, he has taken the differentiated cells from the tadpole. So both of them have seen that after introducing the nuclear material to the enucleated oocyte, it has developed to tadpole. But what is the difference in these two? Where the cells taken from frog embryo or the early stage, most of them developed into tadpole, and here less than 2% develop into tadpole. So what basically it suggests, from at the stage of the development or the stage of the material from where you are taking the nuclear material, whether it is early stage or late stage or younger people or adult, so it makes the difference. The success rate varies with age. So what does it mean? As we age, is it that our stem cell pool get deteriorated or becoming less? And yes, the answer is yes. In fact, as we age, our stem cell population actually is going down. That's why it is suggested you have to keep yourself active with exercise and other aspects. So at least you build up your immune system and your stem cell pool also. To some extent, you try to make them in a continuum state. Next, as we know, the SCNT, this was actually the pioneering work by them, which came into being by the work of Dolly. Now there are many more, Dolly, Molly, Polly, and there are many more organisms. So this is the sheep which has cloned. This is the first one. A group from UK, Ian Wilmot and his associates, they have developed this using the same SCNT strategy, using the enucleated oocyte or the egg and taking the cells from another sheep and then developing this. So this is reproductive cloning to get the sheep Dolly. And then, as I told, if you are restricting at the stage when the blastocyst stage and taking the cells, that is called therapeutic cloning. The next cartoon shows when in for a simple blood test and get cloned by mistake. So this is just to show if by mistake you get cloned, which is ethically incorrect in human case, but in animal case, in the veterinary field, this is in fact true and one can probably do it. So what are the limitations associated with this? Availability of oocytes, and as I told, the ooplasm is still intact and you are getting the genetic material. So it causes the heteroplasmy. And there are something called epigenetic signature when we develop. We have got methylation, demethylation, de novo methylation. So those are the aspects during development, these are actually in place. But when you are trying to rewrap or manipulate the system, sometimes the epigenetic signature is lost. By that also there are a lot of defective tissue types or the defect in the development proceeds. And another limitations in this case, influence of differentiation state. As I showed also the tadpole, part of John Gordon who has done the experiment, and Briggs and King. So there the developmental stage does matter in the success rate of that. Sometimes the cell type from where you are getting the nucleus from or the nuclear material, that also retains the memory, because of which after you develop also embryonic stem cells, there is some retention of the memory, or when you differentiate the cells, they give rise to cells. For example, here you have normal somatic cells. It has come from probably muscle cells. So many of the cells, they can actually, when you differentiate those, they retain the memory of muscle cells, and during differentiation, they go more or directed more towards the muscle cells. So that is the memory of previous gene expression is retained. So this availability of oocytes, people thought how to actually have the oocytes available. We are thinking of using rabbit oocytes as a system to carry out this SCNT process. But now the success rate is so low, people are not going for this SCNT. And especially after the advent of induced pluripotent stem cells, that has drawn much more attention compared to SCNT. So this is another way of getting embryonic stem cells from. First is from the developing embryo, as I mentioned, then SCNT. Next comes parthenogenetic means. Parthenogenesis is the vegetative propagation. In veterinary field, as we know, this is the way many times also they try to develop, because if there is a superior herd, so that's why they want to maintain that particular germplasm and they go for vegetative propagation. So by that also, you can take the oocyte, you can stimulate that and bring back to the blastocyst stage, and this inner cell mass cells you can take and then culture those, develop the embryonic stem cell line for downstream application. Then the next one comes, once you get the embryos, I was telling how you can get the ICM cells to establish the embryonic stem cell. So this is the technology which is used for immunosurgery. I explained already. So this is what I was talking about, the mucilaginous layer, the zona pellucida, that we dissolve with peptidase. Then we use the immunosurgical technique or the antibody followed by complement. We choose up the outermost layer, then you are getting the inner cell mass, and that we culture further. And you can see this is actually coming from the human embryo, which is the work done in our lab. And this is the cell line that has been developed from the ICM cells. And these are grown in a bed of fibroblast feeders. We provide trophic support or the feeder support for the maintenance of these stem cells. And the downwards, these are the mouse embryonic stem cells. Those are grown with fibroblast and these are grown without fibroblast. Mind you, here, these are actually, these whatever you are seeing the colonies, these are coming from only single cell. So what it indicates is single cell is capable of renewing itself and giving rise to this colony-like structure. And these are the cells further you try to propagate and then you make a line. And those also need to be characterized. I will not discuss much on that because of the time constraint. So you try to see whether these are the true embryonic stem cells. So they need to have the specific characteristic features which the pluripotent stem cells should retain. So they are identify specific gene signatures. So we try to see whether these are expressing those genes. Then we try to differentiate whether they maintain the pluripotent nature. That means they are giving rise to all the three germline derivatives. I'll show you also in subsequent slides how the pluripotent signature we can identify. So before saying that, the next is induced pluripotent stem cell. From there also you can derive the stem cells, embryonic stem cells or pluripotent stem cells from. So I was talking about the Nobel Prize or the Nobel laureate Shinya Yamanaka's talk. So he is the one, the Yamanaka factors, you can see in the first one, OCT4, SOX2, KLF or CMYC. So he has actually screened 24 such factors and nailed down to only these 4, and which are called as Yamanaka factor. So what he has done, he has taken the fibroblast cells and introduced these specific 4 factors, and then he has got the colony like what I showed the previous slide, the colony like structure, and those are the embryonic stem cell like cells or induced pluripotent stem cells. Further, with the recent advancement, the Yamanaka factor, not only these factors, there are many more other factors have been also introduced into the field, and many more ways to make the induced pluripotent stem cells. So why it is so important? Because in this case, you are taking any somatic cell. So if I need some cells for myself, if I have any degenerative disorder and I want to utilize stem cells for therapy, so I can take any cells from my body, either it will be blood cells or the normal fibroblast cells, then I will make the pluripotent stem cells. Why? Because if these are pluripotent, I can direct those cells to all 3 germ layers or any cells of my interest. If I have some kind of disorder in the, for example, I have some muscle defect from some degeneration, I want to make the muscle cells. So since the cells are coming from me, those will be specific for myself. And using this strategy, I can make the cells of my own. Not only that, this will also serve as a cell type where you can mimic the disease. If I have any genetic defect and if the cells are taken from me, so the cells that I will get in my dish, those will resemble myself with the carrying out the same genetic disorder. So I can simply mimic the disease in a dish. And why it is important? Because most of the cases when you do research, we take animals into consideration for any disease research. In this case, since I will be mimicking the disease in a dish, I don't need actually for proof of principle studies, I don't need to sacrifice animals. So it will be true replica of a disease. And that too, for many diseases, the mouse model or the animal model is not available. So in that case, one can utilize this strategy to make disease in a dish. So these are the different ways and means you can get the embryo or embryonic stem cells for the further applications. In this is the work I am showing the example. We have used normal fibroblast cells from where, as you can see, over a period, so after some time you get a colony of cells. These are the fibroblast cells. You can see those are like fibroblastic morphology or the spindle-like morphology. After a while, after introducing specific factors and specific growth conditions, you can see a colony appearing. Then over a period, you can see these colonies are growing, and then these colonies are taken and further propagated and differentiated. So this is just to show the example how the pluripotent stem cell look or the induced pluripotent stem cell look basically. Now I will come to the adult stem cells. Adult stem cells are tissue restricted, so there also you need to see what are the tissue sources, how will you isolate, how will you maintain, differentiate, and can they show some plasticity, which is coined as transdifferentiation. Then you have to characterize, enrich and purify. So what are the adult stem cells? In our body, as I mentioned earlier, we have distinct population in different organs. We have got adult stem cells. Neural stem cells are there in brain. Bone marrow is the best example which has gone to clinic, which contains 2 types of stem cells. One is hematopoietic, one is mesenchymal stem cells. One can also get from the dental pulp, the stem cells. These are containing the mesenchymal stem cells. Even muscle adipose tissues also contain mesenchymal stem cells. Even from the bones, you can also get the stem cells, which will also have the, and during development when the gamete is formed, that time you are getting the EG or PG cells, primordial germ cells, which are pluripotent. So from different parts of our body. But the question comes, if you require the stem cells, how will you isolate from different organs? Can you really cut open the tissue and get those cells? Because it will be invasive. So bone marrow has gone to the clinic, still it is quite invasive procedure to isolate the bone marrow. So that is why people are going for the fetal stem cells or the ones well thought of waste you are making from the umbilical cord blood, placenta, umbilical cord. Those are rich sources of stem cells, especially the cord and placenta. You can get the mesenchymal cells, even adipose tissues. But from the cord blood, you can get both hematopoietic and mesenchymal. Here I am giving the example, mesenchymal stem cells from different sources. This has come from the human bone marrow. This has come from the umbilical cord blood, umbilical cord and placenta. As you can see morphologically, those are like fibroblastic morphology as spindle shaped, whereas UCB, here we can propagate these cells and we can also differentiate, and these are multipotent in nature in the sense they can give rise to bone, fat and cartilage. So they can give rise to 3 different cell types similar to hematopoietic stem cells which give rise to all the blood cell types. These can give rise to bone, fat and cartilage. That's why those are called multipotent stem cells. So I'm just giving you the example. Depending on what cells you would require, you can get the stem cells from that, but since this is invasive procedure, one probably will not go for cutting open the organ or getting the stem cell, isolate those for further implications. That is the reason why people have taken bone marrow derived, because that is, even though it is invasive, you can get both the cell types, hematopoietic and mesenchymal, for further applications. But now many of the doctors or many clinics are claiming as if bone marrow is the panacea for many of the diseases. That is in fact, even though now the research is going on in understanding stem cell plasticity. What is stem cell plasticity? That shows whether the adult stem cell can be plastic. Instead of giving rise to only a single cell type what they are supposed to or destined to form, can they cross the lineage barrier and give rise to other cell types? For example, can a muscle cell give rise to neural cells and vice versa? Same also blood cell give rise to nerve cells, vice versa. So that is what many people in fact they have shown that this is possible. Still it is controversial. So one can study not only the, and when you isolate these cells, not only you can see how they grow, whether they can grow and differentiate into different cell types, and whether they can retain the plastic nature. So these are the characteristics one has to go for. So once you isolate the stem cell, you have to see whether they are self-renewing. Embryonic stem cell will see whether they can renew indefinitely. And how does it happen? As we know, every cycle of cell division, we have in end of the chromosome we have got telomere. So that's shortness. So because of which after a while, the cell actually there is replicative senescence is attained. In case of embryonic stem cell, there is very high telomerase activity, because of which this replicative senescence doesn't happen in that case. So they divide indefinitely. Whereas adult stem cells, they are limited in their self-renewing ability because of this telomere shortening. Then comes differentiation. I have already discussed about that. The potential of the cells, how can they differentiate into pluripotent cells can give rise to all the 3 germinal derivatives, multipotent, depending on where from they are derived. Hematopoietic, neural, mesenchymal, those are multipotent cells. Unipotent, they can give rise to only one single type, like liver stem cells or skin stem cells. And plasticity, if they can cross the lineage barrier and give rise to another cell type. Then comes regeneration potential, that is a means of repair that is mostly confined to adult stem cells. As we know, liver regenerates. How? Because it has got stem cells which are actually helping in the regeneration potential when there is any kind of damage. Same also, most of the adult stem cells, those are actually capable of regenerating as and when the insult comes. They remain in a quiescent state, but as and when the insult is there, in response to the insult, they come to the mitotic, they attain the mitotic state, and then they try to divide and differentiate. And they are also amenable to genetic manipulation. So these are the specific characteristics one can see after you isolate the stem cells. You have to characterize these by different means. Due to self-renewal, it can be either symmetric or asymmetric or both. So if you come to embryonic stem cell, mostly those are the symmetric because they indefinitely self-renew. So they give rise to them. The stem cell give rise to themselves. One cell give rise to 2, 2 to 4, like that it goes on. When it comes to adult stem cells, these are like in that case it will be adult stem cells. Stem cells they can give rise to progenitor or precursor for particular tissue type. Then it can mature. So during that process, what happens? First, the stem cell gives rise to 2 stem cells. Then each one, it may give rise to 1 stem cell retaining the same parental property. Another one can mature to more differentiated cells. So here it becomes asymmetry. And when the progenitors are formed at this stage, they can give rise to 2 mature cells. This becomes more committed or 2 differentiated cell types. So this is the way the cell renewal proceeds in our body. So this is one way of characterizing. The second, to understand the differentiation potential. How would you understand the differentiation? Because every cell has its own signature, and if you have to differentiate or direct those cells to different cell types, because this is artificial system, when you are isolating the stem cells, you have to see whether those are actually differentiating into the cells of our interest or retaining the differentiation potential. As we know, we require also specific growth factors, or we require nutrients for our survival and growth. Same also is true for any cell types. So if you are culturing stem cells also require specific factors or the cocktail of factors where you can grow those and differentiate. So by manipulating the cultural conditions, one can see what kind of differentiation status they achieve. And once you can manipulate in terms of the not only the growth medium or whatever exogenously added factors you are providing, even what kind of attachment, cell attachment, or the extracellular matrix where cell-cell interaction is taking place, you have to manipulate that. And also, like in case of iPS cells, you are introducing genes, so you can alter the genetic signature. That way also you can manipulate and then understand the differentiation signature or the differentiation status. So this is just like, because this is very coordinated fashion it goes on. You just think of when you are crossing any, like when you are going on the way, we have traffic light or the signals. So there are 3. One is the red one gives right to stop. Then the orange one, it says get ready. Green one, get set go. So the same way in our cellular architecture, this is how it is defined. When embryonic stem cells, you are directing those to differentiate. So that time you have to have some factors coming into play and asking the cells, okay, now you get ready to go towards this particular lineage. So at this cells case, when the orange light comes, the cells are getting ready to give rise to another cell type, and when the green signal means that time some factors come. So using those factors, or those factors are helping the cells to give rise to a particular cell type, or they are destined to give rise to that particular cell type. So it becomes green, and the factors are playing a role to direct the cells to differentiate. So this is a coordinated fashion. So as you can see, the same way, when this coordination is happening, everything goes smooth, and all the different developmental stages or the different tissues, organs are coming, and the entire organism formed. So this is normal way. But if this coordination is lost, what happens? Like you can see in the station or next to the airport, what happens? Because nobody wants to give way to anyone, so it is chaotic. The same, sometimes it also leads into fatality, and there are a lot of disorders. That is what the developmental disorder happens when this coordination is lost during development. So this shows basically everything goes in a very coordinated fashion, and there you can see how the stem cells and the factors or the critical ones which are playing critical role, those are the ones contributing the cells, or they are directing the cells to take different heads based on what kind of signal, the cell-cell communication, what is happening, the crosstalk, that is called crosstalk, and it's basically coming from the cell, the intrinsic genetic material, as well as whatever the cellular crosstalk, how they are paracrine, exocrine, or autocrine as well as paracrine, those ways, how those are influenced, and the cells are directed to go to a specific lineage or specific cell type. So the question comes, in our body, our every cell contains the same genetic material, but why the heart cell function in a different way from the nerve cells? So these are the ways and means, because we know gene signature is same, but still the regulation is different. Not only that, even the epigenetic signature is different. So there are different ways these are actually controlled, and the factors which are binding the cis or transactive factors, which are binding to the cis regulatory elements, they are directing. So there is the genetic way and the epigenetic means. Even how the cell-cell communication is taking place, all these are playing a major role to direct the cells to specific lineage. And this, when the stem cells, as I was mentioning also, the cancer stem cell already I have explained. The normal way, as you can see the hierarchy, is for example hematopoiesis. So the stem cells, it can give rise to the progenitor, either myeloid or erythroid or whatever. Then you can, that can further differentiate into more mature cells of blood lineage. But hematological cancer, what happens? They don't follow the specific or coordinated behavior and they proliferate much more compared to differentiation. That's why it leads to leukemia or other blood-related disorder. So, which is more important, all the factors should come together and they should function in a coordinated fashion so that the development will proceed normally. And same kind of condition also, when in vitro we culture the cells, we provide them the same cocktail of factors by which we can direct the cells to differentiate. So here is the example showing mouse embryonic stem cells you can differentiate into neural lineage, the work from our lab. So these are giving rise to neural stem cells which can further differentiate into astrocytes, oligodendrocytes, neurons and different subtypes of neurons, dopaminergic, serotonergic. So this is showing the multipotent nature of neural stem cells. So these, whatever the red colors and all you are seeing, these are the signature actually which identify the cells as neural stem cell, that is nestin is one of the markers. Same also true for neurons, we have the marker MAP2, TOS1, many other markers. So by that, as we are also known by our names, the same, these cells are also known by their identity factors or the identification factors. Using that, we can understand whether the cells are differentiating into that particular lineage or not. Next, showing the human embryonic stem cell that we have developed. So these are differentiating into MAP2 positive neurons and dopaminergic neurons, which is actually the signature is tyrosine hydroxylase, one of the markers. So we have identified these markers, so showing that cells can differentiate into neural lineage. Then coming, not only understanding that, one can even study how the development or the life one can monitor, how the cell divide. Once you culture the cells, when you look under microscope, we think that they are static, which is not the case. Actually, they are very, very dynamic and you can see how the cells interact. This is actually showing the dopaminergic neuron development from embryonic stem cells. And this is another showing the iPSCs that I showed earlier. Those have been differentiated into neural lineage. Not only these are ectodermal derivatives, what I showed of the neural cells. You can see also the mesodermal derivatives. You can see the pulsating structures. Those are the cardiomyocytes, which have been differentiated from embryonic stem cells. And you can see also how the synchrony is maintained. The wave-like pattern is actually, you can see the contraction and expansion, how the muscle, it takes place in the cardiac cell. The same way it is maintained, and they are maintaining the normal physiology of cardiomyocytes. Next, I will show you the skeletal muscle cells. So these are actually basic examples. After getting the stem cells, you differentiate into different cell types and get the cells of your interest. These are done by cultural manipulation, giving specific factors in the culture medium and directing those cells to the cells of our interest. This is showing the pancreatic beta cells coming from the stem cells. And they in fact show the glucose stimulated insulin release, signifying that these are physiologically active. Same also when we have done dopaminergic neurons, we have seen those cells are secreting dopamine, showing the physiological relevance of that. And this is a compendium slide showing the embryonic stem cell differentiating into ectoderm, mesoderm and endoderm, showing the pluripotency in vitro. In vivo also one can study the pluripotency by injecting the cells to the skid mouse. So this is what you can see. Those are the benign tumor form which are called teratoma. So these teratoma, if you excise and take the sections, one can see all the three genre derivatives. Here we are showing smooth muscle actin, that is the mesodermal derivatives. The beta protein, that is the endodermal derivative. nestin, that is the neuroectodermal derivative. So ectoderm, endoderm and mesoderm. So this is showing the mouse embryonic stem cell pluripotency in vitro. Same also true for when you isolate human embryonic stem cells or iPSCs. One can see in vitro pluripotent nature as well as in vivo. This is actually the gold standard test for in vivo pluripotency test. There are many other ways also one can do that by chimera and other aspects, but this is the much more easier way to show in vivo pluripotency by inducing teratoma. This is another example of adult stem cell, the mesenchymal stem cells as I was showing. Those can also be directed to difference into osteocytes and adipocytes. These osteocytes, those have been stained with alizarin red, showing the mineralized, the calcium deposits. And these are the adipocytes, the lipid forming, you can see. Then comes the plasticity. We saw the differentiation, the intrinsic differentiation potential of embryonic stem cell or adult stem cell, and then comes the plasticity. Can they cross the lineage barrier and give rise to? That also we have studied, even though it is a controversial thing. So we have tried to induce the mesenchymal stem cell into neural-like cells. Providing specific cocktail of factors, we do see the structure similar to neurons or astrocytes. I can show you the video how the cells, actually the mesenchymal stem cells, they are forming neural-like structures. So these, what we have seen, it is a transient phenomenon, but many groups have shown that they are capable of giving rise to neurons, and in vivo also they have shown by implantation or the transplantation they are showing some functional restoration. But still this field remains controversial. Some people think it might be paracrine influence, because of which we are getting the functional restoration. The next slide would show another, these mesenchymal stem cells. Sorry. Here you can see those are giving rise to the blood vessel or the vascular cells. These are the initial stage and they are giving rise to these vascular cells, the tube-like structures. So that shows these cells can have some plastic nature. One probably have to explore more. So in vitro, by giving specific conditions, one can probably direct the cells to the cells of your interest. So either it will be intrinsic differentiation, or you can direct those to the cells to cross the lineage barrier and show plastic nature. Recent studies show even embryonic stem cells, if you can provide specific factors, they can also differentiate into trophoblast-like stem cells. So that means they are crossing the embryo proper, whatever I discussed earlier. They can cross that barrier and give rise to extra embryonic types of cells. So in this context, one can think how narrow the field is. Like in context of the potency, it is becoming much more narrow. So one can probably explore this field in much broader way to get the cells of our interest from any cell type. But that field, there is a long way to go. We have to do much more groundwork. Now I have to come and discuss about why you require stem cells. So it's basically, as I told, depending on the source of derivation, you are getting the stem cell from various sources. So what is the implication? Why you require these stem cells? Here it comes the engineering and modeling of stem cell, EM. From S to EM, these are the E and M aspects. One can understand the basic biology using these stem cells, the intricacy of development. When we are seeing different cell types are coming, so one can understand how every stage in a temporo-spatial manner, cell-cell interaction and a temporal, time-dependent way, how the development proceeds. This is the intricacy of development. And what are the applied aspects? One can actually, for the diagnostic purpose, you can develop disease model, predictive model for drug toxicity. In veterinary field, livestock germplasm improvement, where reproductive cloning can be done. Therapeutic implications, one can have cell replacement therapy in terms of cells, antibodies, proteins can be developed from this, and finally the cell, tissue or organ transplantation. So how will you do the different challenges in front of us? One can prevent that, repair or regenerate. So as we know, liver has intrinsic regeneration potential, but there are many cells that we have to see, we have to exogenously supply the cells. There one has to think what kind of stem cell one has to take, and if there is a genetic defect, one can go for gene therapy, gene-based therapy. which Jim Wilson, he is the pioneer in this gene therapy. And then lost cells you can replenish, cell replacement, you can lost organ tissue engineering or organ transplantation. In that also stem cells have much more potential. So first coming to gene therapy, it's actually you are replenishing the defective gene. But that what we require, you require a delivery system. So you have the gene which is the healthy gene counterpart of the defective gene. Then you have got the vehicle which is basically the viral based vehicle is used. Then you incorporate that and then try to inject. So this can be done either through the stem cell or directly injecting the person. So through stem cell when it is done, so you have the embryonic stem cell, you introduce the gene of interest or iPSC cell, iPSCs, patient specific iPSCs. You have got this cell or adult stem cells, you introduce this, then it can be given to the patient where it has got. And if you have genetic defect, you are taking the cells, develop iPSCs, correct the gene, defective gene, and give. So this way the genetic defect, that's how it can be addressed. Sickle cell anemia, for example, many people are studying that now. Next is cell-based therapy. So as we know that we have in this population the stem cells, but getting a hand on those stem cells in our body is difficult. So we have to think how to actually get, especially from the pluripotent stem cells. If you get patient-specific stem cells or iPSCs, then whether you can differentiate into the cells of your interest wherever the degeneration is taking place, then you can give to the patient. So there one has to think, what are the prerequisites? If you, for example, if you have Parkinson's disease, where you know it is a movement disorder and the dopaminergic neurons are lost. So the dead nerve cells not producing dopamine. So one has to think of taking those cells, implant, then it has to integrate into the host. So new nerve cells will produce dopamine. Then it will transmit the signal because they are neurotransmitter, because of which our movement is controlled. So in this regard, one has to first think what kind of stem cell one will take, which is the right cell. Multipotent, totipotent, pluripotent. Obviously, totipotent cells zygote, nobody, it's ethically also incorrect. Pluripotent, one can think of iPSCs as one of the types. Multipotent, so if you can show cellular plasticity, maybe in that case, but obviously no one will cut upon the brain and take the neural stem cell for that. Then once you take, if it is allogenic source, obviously you have to match so that there is no transplant rejection. Followed by, you have to put also at the right place. Since the dopaminergic neurons we are talking about, so you have to inject or incorporate the cells or transplant the cells at the right place, that is in the brain, in the midbrain region, the striatum region. Then you have to see whether it is integrating and functioning normally. Such studies have been done using the fetal stem cells in the Parkinsonian patient. So, and the cadaveric patient sample. So this is before surgery. Here you can see the DA neurons or dopaminergic neurons are actually lost. That is in the midbrain region. After transplanting, you can see the restoration of the cells. We have also done similar studies in mouse model or the rat model. Injecting the embryonic stem cells at a particular stage. These are embryonic stem cell direct injection and specific. So this is actually hemi-Parkinsonian model. One side we have created, actually destroyed the dopaminergic neurons and transplanted the cells. After transplantation, you can see this is the restoration. So this shows the potential of these cells in cell replenishment or cell replacement therapy. Hematopoietic stem cells, as I told, this is the best example which has gone already to the clinic. Now in limbal stem cells, for the ocular disorders people are doing, and L.V. Prasad Eye Institute is doing wonderful work in this context. Even AIMS and even Chennai, Sankar Netralaya, they are also doing similar studies. And already I have mentioned about the iPSCs, because here you can get any cells from the patient body, make the patient specific stem cells. For any kind of disorder, the same cells can give to the patient or transplant back to the patient without any immunological complication. iPSCs now in Japan people are using for retinal disorders, macular degeneration. Even in UK also some groups they are trying this. These are the cells showing the glomerulus of the kidney cells. Those are actually in animal model, they have shown the transplantation and proper recovery. These have got tremendous implications for the future medicine, the regenerative medicine. But still, we have got a long way to go to understand the basics. Still, I would say that we are at the infancy stage to understand what the stem cells are. Lot of advancement has already been done, but still there is a long way. Actually, this is tissue engineering. I was telling how will you engineer. So it's not only cells. Cells, this is actually, it's given as cells per se. But if you think in terms of any organ degeneration, there is shortage of organs. So one can think of engineering the organ and implanting the same. So how will you do? This is actually interdisciplinary, truly interdisciplinary, combining the principles of engineering and life science. So here what you can do, you can engineer organs or miniature organs, that is called organoid, that can be done to actually understand the development, or disease you can mimic in a dish, a bioimplant, where the implants laid with biological samples or tissues. I will give you some examples of this. So what are the specific requirements? One can think of, okay, if you want to engineer something, because it's a 3D structure, so you have to have a skeleton or the cementing material. So that is called scaffold or biomaterial, biomolecules, which can be natural or synthetic. Then you have to have stem cells or the cells that you want to actually implant there. And this scaffold will have, it will maintain the 3D structure. Then you have to have the stem cells there in the right proportion, right place. And this biomaterial needs to be biocompatible. Those will be non-cytotoxic. So for example, and even in some cases it needs to be biodegradable. Soft tissue basically prefer biodegradability. If it's a hard tissue, for example, bone defect, there you can have the permanent implant, bioimplants are there. So for example, here if you have some chondrocytic defect, osteoarthritis, so you can take the mesenchymal stem cells, so you can have a scaffold, take the MSCs, culture that, and you can implant. So the MSCs can give rise to chondrocytes. Those are the cartilage cells. Using the same strategy, so far we have actually, people have succeeded in developing bioartificial skin, which is very similar to normal skin, vascular prosthesis, nerve guidance channel. And Kasai at Riken, he is the pioneer. And he is no more, unfortunately, an eminent scientist. So he has developed retinal cells. So this is what the news in 2011-12. Retina in a dish, tissue engineering the brain maker. So they have taken these induced pluripotent stem cells, provide a support or the matricial like support, and they have shown in a proper condition. This is actually invaginating and making the optical cup like structure, which is shown here. So in this actually it is having the lens like structure, the retinal pigmental epithelia. So these are the ones which is showing actually or making the eye. So this has gone to the phase 1, phase 2 clinical trial in Japan. Same also is true for making brain, mini brain. So in Austria, one group, Noblich, they have developed from the human embryonic stem cells. Even people have now succeeded in making from iPSCs. These are called brain organoid. You can see the optic cup like structure here, which is showing a black pigmental epithelial cells. So one can get the brain, miniaturized brain from this. Another group also in Ohio State University, Rene Anand, he has also shown the same optic stroke, cephalic picture, and 2 hemispheres of the brain. So why I am showing this, one can actually in vitro manipulate the conditions and the 3D structure it can give, stature it can give, and you can get the miniaturized organ to understand the development of that particular organ or tissue. And this can also serve as a very good model to understand disease per se. It's not only to brain, it has gone even for the kidney miniature, kidney development, heart development. I think I have to probably go faster because the time, I probably have exceeded my time. I'm sorry. I'll just go fast. So this is just to show actually the tabular form where all these organoids have been developed. Recently during this COVID period, people are also, many groups, in fact worldwide, people have done culture from many different tissues, long intestinal tissue, because as we know, this COVID is affecting all the tissue types. So people are studying this COVID infection in different cell types using the organoid model. Now they have succeeded in engineering trachea, which have been implanted to the patient, and they have also followed up studies also show the success. The other advancement which has gone to customized organ. So what they do, they take any cadaveric sample, for example heart. You take that heart, using specific detergent you decellularize. So after decellularization, so because it is having the skeleton already, so you try to inject the cells of interest at the specific place and specific proportion. Then you are getting the entire heart. Then you can give rise to the electrical stimulation for the heart to pump. So this is the recent advancement which has been published in Nature 2013, and this is another example in Texas. They are just waiting for that to make the heart, injecting the cells of interest. The most recent advancement is the 3D printing. So in not only decellularization organoid model, so you can make 3D printed organs per se. So what is the principle? It's like a bioprinting or 3D printing. So as we print also the same way, the different stepwise. First is image. So the cells of the organ that you want to print. So first you have to take the image of that either by X-ray, CT scan or MRI. Then you have to you have to use the computational or mathematical tools. So you have to design how this particular, after taking the picture, you have to design how exactly you have to put everything in place. So once you design, you have to take the material. So what should be the scaffold material, whether it is synthetic polymer, natural polymer, ECM components. If it is hard tissue, you have to also select the material accordingly, cementing ones. Then cells, whether it will be more differentiated cells, pluripotent stem cells like iPSCs or multipotent stem cells. Then once you know what cells you have to take, what scaffold material you have to take, you have to use the printer. Either it will be inkjet, micro extrusion, so it's actually layer by layer it starts printing, or you can also do laser assisted. Once it is done, then you have to see how the cells have grown and then whether you can implant the same. The next also shows the same thing here. If you have decellularized matrix, they are also layer by layer. You can do the printing and get the organ or the tissue structure, whatever in 3D, for other downstream applications. Like last year, one of the German groups, in fact, they have come with another strategy. So you make the organ transparent. To make that, so you can scan this easily, and once this is transparent, so you can take the image and you can know which place what cell types can be, the right proportion cell types can be incorporated. So by that, you can get the organ of interest. This is actually the most recent advancement in the field, and people are also, many groups are trying this way to develop the organs. The other one, which is like theoretically possible, but practically nobody probably would take. It's more ambitious proposition. When all this 3D printing and other things are not there, people are thinking whether you can actually inject the stem cells halfway through gestation in pig or some other species which are more close to human. Then get the offspring containing the human cells. So these cells can further be implanted. And these are called humanized organs, which are done in pig, but there is always the apprehension that it may contain Yersinia virus and all. So nobody probably would prefer. Now this is the era of 3D printing, and one can probably think of developing organoids, that is for basic research, and printing different organs and tissues. This is some examples I am trying to show from our lab. We have also developed specific scaffold material, and we are seeing the bioactivity. This is for bone tissue engineering. I will not go much deeper. It's the electron microscopic image showing the calcium deposit, showing the bioactivity. And these are the titanium scaffold. So we have done in collaboration with SECRI. So those also we have laid with mesenchymal stem cells, which can give rise to osteocytes. And we have shown they do adhere. and differentiated to osteogenic lineage. So not only this, not only organoids, even making babies. This is possible, of course, in we should not think about in human case, but in animals in the veterinary field you can do. So this is showing just the example of mouse. So you can also get, depending on whatever, if you are taking embryonic stem cells or pluripotent stem cells, you can develop into primordial germ cells. Then you can have the specific like stem cells for the ovary or testes. Then you can get those designer babies or the whatever the organism of your interest by giving those to the foster ones. And then when it is fertilized, you are getting the viable embryos and the offsprings. So this is what is shown by one of the groups. From the lab-made eggs from ESCs and IPSC, you are getting the pups. So that is possible in the veterinary field. So these are the actually wide gamut of implications the stem cells can provide us. So this is what I showed, and even in pharma industry, this can serve as the model system for screening and addressing the toxicological uses of the toxicological testing for various drugs before coming to the market. So I have enlisted some of these actually aspects. So that is also possible. So it is from base to bedside and in the pharma industry. So it has got wide gamut of implications. Not only as a basic researcher, we can study the stem cells, we can have therapeutic implications, we can have implications in pharma industry. So this is actually taking into, as I hope that I have convinced you, saying that how the knowledge from different fields can be brought to understand these cells and utilize that for the betterment of the mankind in different fields, right from industry, clinic and basic research. So this is actually the summary slide showing what are the specific implications. Where we are today using different stem cells. So bone marrow is the best example, even the limbal stem cells, which has gone corneal or limbal epithelial stem cells for ocular therapy and restoration of vision. It's the wonderful work being carried out. So what I have shown, manipulate genes, one can do using stem cell genetic engineering, lab grown organs, tissue engineering, drug designing delivery using a model system, therapeutic cloning transgenesis, which is possible in animal or the veterinary field. So we have to now think whether the future is bleak or promising. But there are a lot of things being discussed. So people think that for every disease, probably this cord blood storage is the panacea, or you can take bone marrow derived hematopoietic stem cell and live. But I would rather say we still need to do a lot of work in this regard. We have to understand the basics of stem cells before jumping into a conclusion. We need to do much more groundwork before going to therapy. If it is autologous, there is no issue because there is no immunological complications. Then it is allogenic, one has to be careful. And we cannot think that one single cell or stem cell can give rise to, or when you implant for any kind of brain disorder or muscle disorder, you just give rise to the bone marrow derived stem cell, they can do. Yes, in some cases it may, because there might be some paracrine influence, but still a long way to go, and we have to do a lot of groundwork before coming to any conclusion. So this is my final slide. We need to collaborate because this is an interdisciplinary field. So there should not be collision but collaboration, and in this pandemic period, this collaboration is certainly required, not this, and awareness. So we have to, as researchers and students, future researchers, we have to understand the difference between myths and reality. We have to practice caution before claiming something, yes, this is going to happen. And thank you all. Any questions, I'll be happy to answer.
Thank you very much, ma'am. It was a very nice lecture and nice presentation. You have given very good insight in this particular area. And I think if you allowed, can we conduct this Q&A session later on after this talk sessions?
Sure, sure. Actually, I'm so sorry. When I start talking, I don't realize the time.
So no problem. Because it's very important area, so that we were listening very carefully to your talk. And So we should move ahead with our another speaker, and we will then start the Q&A session later on, because I think we should give time to think to all the participants that they can raise more queries in
Okay,
between.
sure.
Okay,
Sure,
ma'am.
sure.
Thank you.
Thank you so much.
So I request Dr. Sugandha, ma'am, please convene with another.
Yes. Thank you, sir. So once again, a warm thanks to Dr. Nivedita for a very elucidative, very enlightening and informative presentation, ma'am. We extend our heartfelt thanks and appreciation to you. You presented the topic in an extremely interesting way that, we could not see that the time ran so fast. So also your pictorial illustrations were too good and, made the entire talk very interesting. So we are very grateful to you for your time, for your presence, and for your efforts, and making us, understand the ABC of stem cells and the engineering and the implications with potential applications also. So once again, warm thanks to you. Now we'll be
Thank you.
proceeding for, thank you, ma'am. We'll be proceeding for inviting our next eminent speaker for this workshop, and she is Dr. Parul Johari. Let me introduce our eminent speaker to you all. Dr. Parul Johari was through with a PhD in 2019 in biotechnology, where she worked on atom-based protein sequence analysis, that is an in silico approach. She did her MTech in 2008, and she has more than 15 research publications in all reputed journals like Scopus and BMC. She has to her credit 5 book chapters in Springer. She's guided many students for their research projects. Now, she's been working and associated with MIT Institute of Biotechnology at MIT University, Lucknow as assistant professor. She's been working since 2011 till date. And she was also visiting faculty for MTech, that is Food Biotechnology. That was a DBT sponsored course at Institute of Chemical Technology, Mumbai for 2 years in 2010 and 11. She's also been very good at research and has been the co-principal investigator in the DBT project that is entitled as Application of Computational Biology, Big Data Analytics and Management of Healthcare, Drug Discovery and Clinical Trials in 2018 to 2021. That is going to be, I guess the project is running yet. And she's also received DBT CTEP grant for conducting popular lecture series in biotechnology last year. That was in March 2019. We are very happy and delighted to have you amongst us. And now may I hand over the platform to you, ma'am, for your talk. Dr. Parul Johari.
Thank you, ma'am. Thank you so much for the such a beautiful introduction that I have never seen before. So thank you so much, ma'am. Am I audible to everyone?
Yes, ma'am, you are audible.
I would just quickly share my screen. Is my screen visible to everyone?
Yes, ma'am, it's good to go. Just, ma'am, remove that hide option. It is written at the below. Just hide that column. In the last, it
Which
is
one?
showing. No, ma'am, on the presentation slide, just open your slide.
Yes. Okay. Hide.
Hide. Just hide this and yes.
Is my is this Arpit? Arpit, can you just help me out? If you could just let me know whether my audio and video both are coming, or just the audio that is coming, because I could see a video is being crossed off.
Hi, yes, ma'am. Only audio is coming. Just open your slides, ma'am.
Yeah.
Yes, ma'am.
Okay. So I'll just quickly start with my presentation, and before starting my presentation, I must confess that the earlier presentation that was given by Dr. Nivedita, that was really beautiful, and it was very informative. And the confession is like in the initial presentation, it was so dynamic, it was so lively. You can see cells moving, you can see cells differentiating. There were so colorful images, and the pictorial presentation was there, and it was very lively. And I personally believe everyone would have catch off the presentation because it was so beautifully presented also. But the confession is that my presentation would be a very dry one, is because I would be dealing with the in silico aspect. That means the bioinformatics aspect of stem cells biology. So nowhere you will be seeing any live experiments. There will be no mouse, no human, nothing would be there. There will be no moving cells, no tissue, no cultures, only the data. See, in bioinformatics, we people are dealing only with the data and the analysis of the data. So my presentation, before starting, I must say that it would be a dry one because it is dry in silico biology. So this is Dr. Parul from Amity University, Amity Institute of Biotechnology, Lucknow campus, and I will be giving my presentation on computational tools for stem cells biology. So what exactly are stem cells and how they differentiate and where they are being used? This is already being introduced to you in a very beautiful manner in the last presentation. So I will be starting with the in silico aspect, like how the data is being analyzed once it is being generated using your stem cells biology. So I'll just start it off. So quickly, because this is based on stem cells, so I'll just in one single slide, I'm going to just explain what are stem cells, and because the last presentation was very self-explanatory, so I don't think very much in depth is required in this slide. So stem cells are undifferentiated cells that have potential to differentiate into specialized cells. They are important due to their potentials in tissue engineering, regenerative medicines, and as in the last presentation it is said for 3D printing as well. At the beginning of stem cells biology was an experimental subject. Initially, it was just an experimental subject. The steady advances in the refining and extending the experimental approaches have transformed the field rapidly. Recent high throughput techniques like the microarray, the SAGE analysis, recent high throughput techniques have generated large volume of data on stem cells. Generation of these large volume of multidimensional data requires capturing, handling, integration, and analysis of all the data generated in a consistent way to provide new and deeper insights about the underlying mechanism, which is an essential task of bioinformatics. So in bioinformatics, we are basically dealing with the large amount of data that is being developed in stem cells biological experiments in wet lab. More recently, computational approaches have been critical in the translation of high throughput data called as stem cell bioinformatics. Now we have a separate branch called as stem cell bioinformatics, in which the data that is being generated in stem cells experiments that is being analyzed. So stem cell bioinformatics is immersed with the deals with developing databases, algorithms, tools, softwares, visualizing platforms, modeling of the systems, system level aspects of stem cell, and high throughput molecular data. In this present session, various bioinformatics approaches will be discussed. Those are used for making sense out of the stem cells related data that is being generated, analysis of that data, using statistical techniques, and mathematical modeling of that particular data. In the past years, the difference between the pluripotent stem cell and the differentiated cells has triggered a great deal of interest. It is at the center of the basic research of developmental biology. At the same time, there are a multitude of potential applications in regenerative medicines and beyond. Moreover, the existing progress has been made in the in vitro control of basic cellular phenotypes. Finally, the computational methods, that is the in silico approaches, such as the hierarchical clustering, the principal component analysis, what we say as PCA, have been used to investigate the pluripotency and the differentiation in systematic ways, often based on the differential gene expression data computational biological tools play a very important role in the study of developmental and stem cell biology. The high-throughput data facilitate the investigation of the basis of the pluripotency and gene expression profile of embryonic stem cell. So in this particular slide, we are just going to discuss that what are the basic methods, or what are the basic algorithms or the softwares or the analysis ways in which the data that is being generated is being analyzed in silico. So the very first technique or the very first approach we use is HCL. Can you see the image A over here? This is HCL. HCL stands for hierarchical clustering. Hierarchy means there is an hierarchy. These A and B have a root node over here. The C is more close to A and B as compared to D, E, and F. So what is hierarchical clustering? Hierarchical clustering method is a popular method to identify the functional groups of genes and groups data into tree. From that shown in the relationship amongst the data set. The average of this method that it is simple, the advantage of this method is that it is very simple and the result can be easily visualized. As you can see in this pictorial presentation, it is self-explanatory that A and B are more closely related and C is a counterpart of A and B. It belongs to this particular group. So this is called as hierarchy. The height of these bars, these standing bars, the height of these standing bars, these are on a particular scale, which tells you that how many million years ago they were having a common ancestor. Or it could be on the number of genetic variations also. So hierarchical clustering algorithm is based on agglomerative and divisive approach. Agglomerative means we are going to summing up. And divisive means that initially we were having only one thing and then we have started dividing. And agglomerative is vice versa of divisive. Agglomerative means from the scratch, wherever we are getting similarity, we are combining those sets and then we are going to superior orders. That was about your hierarchical clustering. Okay. Next is in this B diagram, you can see there is K-means clustering. K-means clustering. This is also a method of clustering. So the aim of K-means clustering approach is to group data into known numbers of cluster. So known number of cluster means that you should have a prior knowledge. That means in how many clusters or how many groups you are going to basically divide your data. So K-means clustering approach is grouping the data into the known numbers of cluster. If there is an advanced knowledge about the number of clusters that should be represented in the data, K-means clustering is a good alternative. So we can apply K-means clustering only and only when we have a pretty good idea, like in how many groups it is going to be clustered. So it is a good alternative over the hierarchical clustering, and the steps involved in K-means clustering in this particular approach, you can see there are 3. K is equals to 3. 3 is the value of n. So whatever you are writing the value of n, that means in those many clusters the data is going to divide. If you write K is equals to 9, then you will be having 9 clusters over here. Right now in this particular exemplification, we have written K is equals to 3. That means we just need 3 different clusters in which the entire data would be segregated. So the basic steps involved in K-means clustering is, the first step is randomly group the objects into predefined numbers of clusters. So we first define the value of n, that is the predefined number of clusters. Then a centroid is calculated for each cluster, which is used to measure the distance between the cluster. The center value, centroid, is being identified for each cluster. The third step for K-means clustering is that each individual data point is reattributed from one cluster to another, that which depends upon the centroid, whichever the two centroids are closer to each other, they are being clubbed together. The fourth and the last step in K-means clustering is this approach is repeated. That means the centroid, the distance between the centroids is being calculated, and whichever two centroids are close together, they are being merged. And this process is repeated in a manner to fix a number of times. Number of times it is being repeated unless and until we get to the value of n. This is called as K-means clustering. The next approach is PCA. PCA is principal component analysis. So PCA is basically an approach wherein like if I give you an example, PCA is an approach wherein If I give you an example, like if I say that we have got two variables, okay, if I say we have got two variables and I want you to plot a graph, so you can plot a graph on a graph paper because you have two variables. Quickly, you are going to draw an x-axis and a y-axis and you will plot the data. Now, similarly, if I say that we have got three variables, so you will say that because we are having three variables, we need a 3D space, and we will take an x, y, and z axis, and we will take a three-dimensional space and we will plot the data. Okay, now if I say that we have a data which have like 7 dimensions, we have 7 variables in which the data is being scattered. So is it possible for us to even visualize 7 planes? See, if it is 2D, we can put it down in a graph paper. If it is 3D, we can put it down in 3-dimensional. But beyond 3D, it is really very difficult for all of us to even imagine. So how are we going to analyze this type of data? So in this type of data, if we have more than 3 dimensions, we go for principal component analysis, in which we are going to identify those 2 or 3 dimensions in which the maximum scattering of the data is there. So once we have identified those dimensions wherein we have the maximum scattering, that is called as PC1 and PC2. PC1 is always 90 degree to PC2, and we take only those two variations, those two variables, and we plot the data and we perform the clustering. So PCA is basically a dimensionality reduction technique. Dimensionality reduction technique means our human brain is not able to visualize more than 3 dimensions. So what we are doing is we are converting those 7-dimension data, because we are having 7 variables, we were having 7 dimension of data. We are converting it into a reasonable number of dimensions wherein we can plot and finally we can analyze the data. Okay. A very good example of PCA is like if you have a 3D figure and you want to convert it into a 2D figure. So what you do is you put a white canvas at the back of the 3D figure and you throw light from in front of the figure. So what happens? You will get the projections of the image on that white background. So that projections would be the conversion of 3D figure into a 2D figure. So this is just an example like how the dimensions are being reduced. So we basically do dimensionality reduction in PCA so that we can see the data and we can do the analysis part. So this was about PCA. See, these techniques are not limited to your stem cell only. PCA is being rigorously applied in metabonomics. It is rigorously applied in microarray technology. So this is one of the very important dimensionality reduction technique. The other dimensionality reduction techniques are multidimensional scaling, but in stem cells, we apply only PCA. PCA is more approachable in stem cells. So if you could see the third figure is of differential analysis. So the level of expression is being depicted in the color, and a differential analysis aims to identify genes differentially expressed between the distinct group using approaches that account for the typical large number of statistical tests being performed. So whatever are the statistical tests, those are being put together to study the expression analysis. So that was about the part D. Then we have an enrichment, the enrichment zone area. So enrichment analysis, the gene set analysis or GSA, it uses programs such as GSEA. We have programs for this that determines whether the expression of predefined sets of genes tends to cluster towards the top or bottom of the ranked list of the genes analyzed. The analysis is being typically based on differential expression between the two conditions. So it basically gives you the enrichment. This is on the y-axis, you have the enrichment score, and it tells you whether the clustering is being done at the top or it is being done at the bottom. And the basis of that, we are going to analyze the data, or we are going to give the answers. The F1, the last in this particular picture, is mutation calling. Mutation calling is, that means if we have sample 1, the sequence of sample 1, and we have the sequence of sample 2, at a particular position, we are like, if this is my position of reference, wherein I have, this is my position of reference. So in all the sequences of sample 1 as well as sample 2, we are going to compare the sequence of the sequence. And on the basis of that, we are going to tell that what is the mutational rate. Okay, how many times a mutation has occurred? This could also be given in probability or in percentage. So the identification of genetic difference between the samples, maybe from an individual gamete or from a tumor cell or from a normal or a tumor cell, can be compared with an inference within a reference to a genomic sequence, from a normal one to a tumor one, from a one species to another species. So if we are, see, the position is important and the type of change is also important. So whether A is getting transited to G or G is getting transformed to C, so that change is also important, and the position where this change is being studied, that is also important. This is called as mutation calling. And the last one we can see is the peak comparison. In the peak comparison, we have two samples. One is from condition 1 and the other is from condition 2, and we are going to compare the peak. So this peak comparison is to identify genomic loci that are enriched with next generation sequencing, NGS sequencing, that read that the reads have been obtained from the ChIP-seq or DNA-seq or some other calling sequencing methodology. So DNA-seq could be also there. ChIP-seq. ChIP-seq is a chromatin immunoprecipitation, wherein we basically study the chromatin and we study the protein DNA interaction. So this peak comparison can also be done. So this is also one of the very important aspect when we are analyzing the stem cell data. So altogether, in this particular slide, we have dealt with what are the various types of in silico analysis we can do on the stem cells data that are being derived. One is the hierarchical clustering, the k-means clustering, principal component analysis, differential analysis, enrichment zone identification, mutation calling, and peak comparison between the two samples. In this particular slide, they are saying that the integration, none of these science runs on their own. So it's not like that this is one science, or we say that metabonomics is one science that is running on its own. So every science herein are being integrated. So if you could see the image, there are so many sciences which are integrated to your basic stem cell biology or stem cell research. So the inner circle, what you see is your stem cell research, wherein the basic of stem cells and how the stem cells are there and how they are being differentiated and how they are being divided and then targeted. So everything is being written there. And our outer circle, which you see over here, these are the other sections of the sciences which are actually in sync to your stem cell research. Like if we say cell engineering, as said in the last presentation also, cell engineering, or you can say we engineer the cells. If there is some problem, we target the stem cells and we do cell engineering, so that if a disease is being caused because of that particular cell, that is no more being caused. So cell engineering is an integrated portion of your stem cell research. Then you have PPI. PPI is protein-protein interaction, how one protein is going to interact with another protein. So that is also, we have computational protein-protein interaction methods, and we do have the wet lab experimental methods. Like wet lab experimental methods, we have Y2H, yeast-to hybrid system. And in computational methods, we have phylogenetic analysis, we have Bayes theorem. On the basis of that, we identify PPI interaction. One of the very good, freely available online databases as a software is your string database. If anyone over here is interested, they can just log on to the string database, and various protein-protein interactions for maximum model organism is present over there. You can even do the clustering in string database. You can download the cluster, and the files could be uploaded in a freely available software called as Cytoscape, wherein you can find out the hub gene through which the networking is actually happening. Then you have a ChIP-seq, that is chromatin immunoprecipitation. ChIP-seq is a basic method that is used for your protein DNA interaction, because in ChIP-seq we basically precipitate the DNA when it is binded to histone proteins. So the area where this DNA and protein histone are interacting, that is the portion of protein DNA interaction. We have RNA-seq, RNA sequencing area, that is also integrated to your stem cell research. So integration of high throughput data in stem cell research. So all these are high throughput data. I hope you all understand high throughput data. High throughput data means with a single experimentation, you are going to produce n number of data. So in order to achieve better understanding on self-renewal, reprogramming, direct conversion, and other processes in stem cell biology, genome-wide profiling have been conducted at the endpoints, and sometimes during those processes, various types of high-throughput data are also being engaged. So methylation microarray, your miRNA sequencing, then your network. Network means how everything is working inside a cell. So network means it's a working inside a system. So in physics, we used to study now open system and closed system. So cell is a very closed system. Very few techniques are there which are working as an open system in a cell. So how everything is actually happening in collaboration with other networks. So that is your network. So I just wanted to say that stem cell research is not being done as a standalone technique or a standalone science. We have so many integrated sciences that are dealing with stem cells, and one of them is bioinformatics, and that's why we are in an era wherein we are studying stem cell bioinformatics. So wet lab aspect was being beautifully dealt by ma'am in the last presentation. So I thought that, and my zone is also bioinformatics, so I thought that I'll deal with only the in silico aspect. Then these are some machine learning classifiers. So machine learning is basically a technique in which you actually tune the machine. That you tune machine in the sense is, if I say machine, I mean a system. So machine learning classifiers are support vector machines that we called as SVM. So what happens in SVM? First, I'll just let you know what are machine learning, what is machine learning, and what are machine learning classifiers that are used in stem cell research. So machine learning is basically we are training the computer, or we are training the software or the algorithm what we have developed with some known data for which the results is being known to us. And then we test it, that how good, this is a sort of artificial intelligence. We first train the system, okay, if this type of data is coming, then this is the possible result. If this type of data with various, the bigger is the data set, the good is the machine learning technique. So various type of data sets are being used, and they are being fed to the system, and the system is being trained that how are you going to interpret it, how are you going to analyze it, and what would be your depiction on the data. And then we send some test samples for which we actually test whether the machine is giving the correct answer or not. So that all these testing criteria are being, or bioinformaticians are there, they are actually doing it very, very beautifully. And the first one is support vector machine. So first classifier is support vector machine, or the SVM. So what does actually SVM does is it basically divides the two data sets into two different zones. So the red dots you could see over here are the red blood cells, or the RBCs, and the green one is the hematopoietic stem cells, or HSC. So SVM is going to draw a plane. That plane is not necessarily going to be a straight or a parabolic or a hyperbolic curve. It could be any way. See, you can see over here this curve has taken a very different shape. But this curve would be in such a way that both the RBCs and the HSCs are being very well differentiated. So see, in this particular diagram, I have purposely put red blood cells in red color and hematopoietic cells in green color. But when the cells are there, we cannot see them, and we cannot visualize that where we have the red blood cells and where we have the hematopoietic cells. But this algorithm, this machine learning classifier, basically classify the two sections into two separate zones. So this is one classifier. Then you have Naive-Bayes classifier. This is also based on your Bayes theorem, and density is there, and expression of the genes is there, and the two genes are being expressed in two different colors. And the last one is the random forest technique, or the random forest classifier. This is also a classification technique. Along with classification, it does the regression also. So classification means it is going to classify into different sets, and regression means it is going to put a very defined order also, that how the classification has actually been achieved. see, if I say that x and y are both of them, if two variables are there, x and y, and both of them are positively correlated, that means if x is increasing, y is also increasing, or if x is decreasing, y is also decreasing. That means both the variables are behaving in a similar pattern or a similar manner. But if I say x equals to 2y, and if I say x equals to y square, so x equals to 2y and x equals to y square, in both the cases, x and y are going to be positively related, because the way x is going to increase, on the contrary, y is also going to increase. But in the first equation, x equals to 2y, it is going to increase in a linear manner. But in the second one, whereas in the second one, if I say x equals to y square, no doubt x and y both are going to increase, but the increase would be there in a quadratic manner. So it would be a non-linear increment. So positive correlation is there, but the way it is being correlated would be different. So x and y, so this is called as a regression, fitting an equation, exact equation, how the increment is actually taking place, or how the decrement is actually taking place, or if x is increasing, y is decreasing. Or the fourth case is that if y is increasing, x is decreasing. So if we fit in the exact equation, we get the exact pattern how they are being performing. So that is called as a regression analysis, and random forest technique basically deals with the regression. It does the classification, but it does the regression analysis. So now quickly I will just start these techniques. So in this particular section, we have computational tools to address the three major barriers to achieve self-fate engineering. So the three barriers, what they are saying over here is that self-fate engineering, for example, the directed differentiation of PSCs, or the direct conversion among somatic cell types, that is the conversion of fibroblasts to cardiomyocytes, through the epitopic expression of various genes, is practiced in thousands of laboratories worldwide to model organisms, to explore the inaccessible time points in development and the screening of the drugs, and to develop regenerative medicines and therapies. However, there are three classes of barriers that are impeded in self-fate engineering from fulfilling its promise to broadly transform the biomedicine enterprises. And the first class of the barrier you can see over here is, the first class of the barrier concerns the absence of rational, proven, and hypothesis-driven systems to select the conditions to guide the directed differentiation, or to select the differentiated regions. So that is your first case. The directed differentiation methods inspired by our understanding or by our knowledge of signaling, and that is being forced in mouse development, and are limited to our inability to study their transient and their embryonic states. By contrast, the methods to select master regulators for the use in the direct conversions are based on the assumptions of kernel, that is your gene regulatory network, that is GRN, that comprises of your TF factors. And the second one, while the strategies to identify the use of TFs of a kernel GRN was successful in reprogramming back of pluripotency, the extent to which it applies to other cell types is unknown. We refer this particular question, if this particular question is there, we say that it is improvement problems. The improvement problem is that we are not able to differentiate the direct differentiation or the direct conversion of the fibroblast to engineered cell. So if you could see that the improvement problem, which I've just quoted, for this particular problem, these are the, in this particular section, can you see, these are the softwares that are going to deal with your improvement problem. CellNet is there, Crespo is there, Davis is there. So these are the software which are going to deal with your improvement problem. The second one, the second class of barriers concerns to our limited ability to access the comprehensively the fidelity with which the engineered cells resemble their in vivo counterparts. We refer to this set of particular question or this particular problem as assessment problem. So assessment problem means we are not able to match whether it is an engineered cell or a target cell type. And the computational resources to address the assessment problem is the best one is the scorecard. You have pluritest also, you have cellnet also, and you have key genes also. Okay. And the last one is the lineage bias problem. So the lineage bias problem is the third type of major barrier to achieve a self-fate engineering. And the third class of the barriers concerns with the long observed variability between the PSC lines in the efficiency and fidelity with which they can be guided to select the lineage, which we refer to as the lineage-based problem. So this lineage bias problem. So this lineage bias problem in this particular problem is being addressed by your scorecards. The only tool available for that is a scorecard. The predicted lineage bias towards the endoderm, mesoderm, ectoderm, and neural and hematopoietic lineage. This is the only software or the tool present for guiding or analyzing or just finding out the answer for this particular type of bias. So I have just in the next figure, if someone is interested, they can take my PPT also. The computational stem cell biology. So all the methods, the types which are being used, the input types, the input data it takes, the species it has been tested with, the cells and the type of tissues it is being tested on, the output it is giving, if at all it is extensible or known, and the resource is being listed. A number of, scorecard is also there, pluritest is also there, cellnet is also there. Okay, so these are just the list and the details is being given over here. The last portion of this presentation will deal with the single cell omics. So single omics, I hope you all understand. We have a variety of omics, like we have a variety of omics sections. So omics is basically we deal with proteomics. So proteomics is dealing with the protein complement of the cell. We have genomics that is dealing with the DNA complement of a particular cell. We have metabonomics. So metabonomics is basically dealing with the metabolites, the level they are being expressed in a particular diseased order or normal order. So we have metabonomics or metabolomics also. We have transcriptomics also, which deals with the transcript, the study of the transcript or the study that is being done at the RNA level is called as transcriptomics. And now we have single cell omics. So single cell omics is the era right now working with. Although there has been rapidly widening interest in using single cell profile across a range of applications, from cancer heterogeneity to the identification of new cell types, and commitment of various reviewers on the molecular biological techniques, the computational side of the appropriately handling these data has received relatively less attention. So here we explore the type of questions that could be addressed with single cell omics, some of the analytical approaches that have been brought to bear so far, and we end up the discussion of the areas where analytics needs to be improved to handle and take advantage of your different types of data sets. So single cell omics basically deals with transcription heterogeneity in stem cells. So what is the heterogeneity? What is the variations that is there at the transcription level of stem cells? Then a population subculture in stem cells. So the population, this single cell omics is especially valuable in stem cells investigation because they can be used to further refine the subpopulation that contains stem properties. Okay, just to find out or refine the subpopulation that contains stem properties. For instance, yes.
Can we go fast, actually?
Yeah, sure, sir. So the next is construction of novel gene regulatory networks. So single cell analysis makes it possible to infer regulatory relationships between genes that were obscured in a bulky way, and then lineage trajectories. So we can introduce a single cell omics to study the lineage trajectories also. So lineage trajectories is basically examining the cell states through development or in the time course. Experiments are especially informed because they can help to reconstruct the lineage hierarchies. And the last one is the spatially resolved single cell genomics. So spatially resolved single cell genomics is fluorescent in situ RNA sequencing, and it quantitates hundreds to thousands of RNA in situ in fixed cells, in tissue sections, and in whole mount embryos also. So these are the areas where single cell omics could be a great advantage. And my concluding remarks for the session is that the emergence of the computational stem cell biology resembles the transition from cancer biology to cancer genomics, where big data, especially the whole genome sequences, required the recruitment and training of the computational biologist or bioinformatician specifically focused on the cancer biology. The stem cell biology now faces a similar inflection point where the emergence of both do-it-yourself or analyze the data is being there. So we can propose that the subdiscipline of computational stem cell biology will yield the transformative insights into developmental biology and will enhance our ability to engineer cells' fate and fidelity and efficiency by synthesizing single cell data with predicted modeling approaches. So modeling is the basic concept of bioinformatics. And finally, I must state that yet there are many open challenges. We anticipate that the computational stem cell biology will produce a quantitative basis to define cell type, identity, and will define the molecular logic of lineage commitment and maturation. Now, finally, the last point which I always conclude in my presentation is bioinformatics is very important for analyzing the data. The correct way, the correct technology, you can analyze the data in all the way you want. It basically depends from person to person and their perspective. So the correct interpretation and the correct logic behind that is required. So just analyzing the data and giving answer is not enough. We have to justify, we have to actually support it with our literature work, with our actually wet lab experiments data. So I end up my session over here. Thank you so much.
Thank you very much, Dr. Parul, for this nice insight and the applications of bioinformatics. I can say there is a vast ability of this application of this area. Conduct in silico work on stem cell. So I just request you please be with us because we will conduct a open discussion with our other speakers, eminent speakers. So please join that session, and we're just moving towards to start the last lecture of this particular workshop with Dr. Deepa Subramaniam, because we are already late, and so just we want to conduct the particular talk of Dr. Subramaniam firstly. So with these words, I just would like to introduce our next eminent speaker, Dr. Deepa Subramaniam. Dr. Deepa Subramaniam is working on cellular trafficking that differ between embryonic stem cells and differentiated cells. So before that, I just want to give her related background, educational background. She has completed PhD from NCC, National Center for Biological Sciences, TIFR, Bangalore. And before that, she has completed BSc from PhD College of Arts and Science, Coimbatore. She is having research experience as scientist in National Center for Cell Sciences from 2012 to present. And she has done postdoctoral from University of California at San Francisco. She has received funding from Wellcome Trust, DBT India Alliance, and also she is having NCCS intramural funds to conduct various research activities. And she is a collaborator with ICER Pune, and she has been published so many research papers in national and international journals reputed. And actually, sorry, yeah. So with this word, I just would like to invite Dr. Deepa Subramaniam. Please give this particular talk because we are waiting for your talk. So we are here to listen you briefly. So now I'm handing over mic to Dr. Deepa. Please start
Thank you.
your. Yeah.
Can you hear me?
Yeah, you are audible now.
And you can see my slides?
Yeah. Please put
Okay,
on full.
excellent. It is on full screen.
Okay. No, it's not full screen right now.
Oh,
Yeah. You just click on full screen icon. It is on below third. Yeah.
now is it okay? Hello?
No, it's not here. Actually, it's Arpit. You are able to see full screen?
Sir, actually, it's not in the full screen mode. It's in the default mode.
Okay, let me try again because I did it.
Yeah.
So I went here to application window and I said share. Now is it full screen?
It is not going to full screen mode. You are just clicking on that icon, but it is not going towards full screen actually.
Okay, because on my computer it's showing it as full screen.
Okay. So can
Are you
we?
using Mac? Are you
Yeah.
using Mac?
Yes,
That's
Nivedita.
problem comes. So I think that is the problem.
Okay, I will
Okay.
do one thing. I will just give me a second. I'll just show it as a full thing and we'll see how that goes. Yeah, I'm very sorry for this.
No problem.
So,
Yeah.
so you can see my full slide now?
Yeah, you just drag that zoom where we zoom the particular slide. Yeah. Just put on this mode and yes, move
Is
on.
this fine? Is this fine?
Yeah. It will be fine, ma'am. No problem. We will
Okay,
attend it.
great. Sorry, I'm very sorry for that. Thank you so
Also,
much.
ma'am, please also cross that slider on which the slides are showing there. Actually,
Yeah.
please cross it. Yeah, now it's fine.
Yeah. Okay. So thank you very much for such a kind invitation and for the introduction. I apologize for the technical glitches. So what I'm going to talk about today is, some work going on in the lab looking at trafficking in embryonic stem cells. And so, let's start by looking at, Nivedita has given a fantastic, start to this whole series. And, this is going to be a tough act to follow up, but I'll try my best. So what I'm going to, sort of talk to you about is, the first few days of life and where exactly are stem cells derived from. And so if you can see this movie, you can appreciate. So this is a movie of a zygote soon after fertilization. So this is a human zygote. And what you can see is that the single cell zygote, it divides first into 2 cells, then into 4, then into 8, and then into 16. And it gives rise to this structure which arranges itself around a central cavity. And this structure is called the blastocyst. And for mammalian development, this is one of the most important structures in development. And this particular ball of cells, which is called the inner cell mass, is what gives rise to the entire organism, that is all of us who are attending this talk right now. So why is it important to look at this ball of cells? Why is it important to study the cells in the inner cell mass? So these are the cells which give rise to the entire organism. That being said, that becomes difficult to study, but it doesn't matter what type of mammal you look at, but that little structure basically gives rise to an entire animal. It could be a mouse or it could be a human being. So therefore, the clues to forming the body plan and for developmental biology lie within this structure, which is called the blastocyst, and which is why we focus our energy on understanding regulation of cells and their differentiation, which starts with the blastocyst. So Nivedita has given a wonderful introduction about stem cells, and Dr. Jory also followed it up with a lot of detail about bioinformatic approaches to study stem cells. But mouse embryonic stem cells are the model system which we look at, and these are derived from the inner cell mass of the blastocyst. These cells can be taken out and put in culture. They have 2 main properties. One is self-renewal, which means that these cells can give rise to more cells just like themselves. And they can also differentiate in response to specific signals to give rise to cells which form the mesoderm, the ectoderm, or the endoderm, the three germ layers. Now, a lot of information has been sort of accumulated over the years, studying what controls the, embryonic stem cell pluripotency. And these involve, molecules such as the chromatin modifiers, transcription factors, non-coding RNAs, as well as cell signaling. But what has become apparent over the last few years is that the process of intracellular trafficking or endocytosis is also very important in regulating the role of embryonic stem cell pluripotent. So what is endocytosis, or what is intracellular trafficking? This is nothing but the process of taking in molecules which are present outside the cell, and with the help of membrane bound vesicles, transporting these molecules inside the cell, which is why it is called transport, right? That's why the whole process is referred to as cellular trafficking. And this happens with the help of certain molecules which help to build a vesicle, which is bound by a part of the membrane. Okay, so in case you were wondering that the cell, or you were imagining that the cell is a static place, let me show you a few videos. I'm sorry, they look very pixelated, but this is showing you the trafficking of molecules inside cells. This is the trafficking of clathrin-coated vesicles which are inside the cell. And as you can appreciate, there's a huge amount of movement of these molecules from outside to inside. Okay. So let me go into a little bit more detail of the endocytic pathway. So let's give names to molecules. Let's specify certain compartments. So if you had a molecule, let's say a ligand, and that bound to a receptor which was attached to the membrane, this could then be taken into a vesicle with the help of certain specific coat proteins, and this would then fuse with the early endosome. Okay, so this endosome is sort of like a head post office where all the molecules end up, which are taken up from the membrane. And molecules which are present here can have two destinies. One is they can be recycled back to the membrane via a pathway involving the recycling endosome, or they can be targeted for degradation in the lysosome through these multivesicular bodies. So I've still not named any molecules, but I've given you a brief idea of what the root is of the motion of the molecules. So the question that we wanted to ask in the lab was, if endocytosis is in some way involved in regulating the pluripotency of stem cells, can perturbation or disruption of the endocytic genes affect the pluripotency of embryonic stem cells? And to do this, basically, we conducted an siRNA screen to identify components of the trafficking machinery which regulate pluripotency. What did we do? We plated embryonic stem cells in culture plates, in 96 well culture plates, and each well received an siRNA, a cocktail of siRNAs which targeted a specific gene. And then we asked, what was the effect of knocking down the specific gene on the stem cell characteristics or properties of these cells? How did we assess the stem cell properties? We assessed it by staining these cells with a marker called alkaline phosphatase. And the idea was that stem cells would have a lot of alkaline phosphatase, a reduction in stem cell properties which cause a decrease in alkaline phosphatase activity or presence. And if we converted these cells into super stem cells, they would have even higher alkaline phosphatase activity. Now, just to make sure we're all on the same page, RNA interference or small interfering RNAs basically act to destroy mRNAs which are produced, and it works, in a way to break down the mRNA for a specific gene through sequence specificity. And this technique the importance of this technique was recognized and was awarded the Nobel Prize many years ago. So briefly, how does RNA interference work? You have a piece of double-stranded RNA, which is then cut into smaller fragments of double-stranded RNA, and the fragments can bind to another protein complex called RISC, which then recognizes the mRNA molecule to which this small piece of siRNA has complementarity and causes the breaking up and silencing of the mRNA so that no protein is formed. So when we did this screen, we screened 112 genes and we knocked them down, and we found that one particular gene, when we knocked it down, it caused a reduction in embryonic stem cell pluripotency. So suffice it to say that anything which lies between these two lines are similar to the control. Anything which lies below these two lines and is marked in blue basically indicates that knockdown of this gene results in a reduction of stem cell properties. So we decided to follow up with this particular gene called CLTC, or the clathrin heavy chain. So what exactly is CLTC and what is the clathrin heavy chain? So clathrin is a molecule which is involved in a particular type of trafficking called clathrin-mediated endocytosis. How does this work? Clathrin assembles a pit around a region of the membrane which needs to be internalized. So suppose you want to internalize this particular molecule, one would then assemble clathrin around this region of the membrane, and along with the help of some other molecules, this results in a deformation and a release or a scission or a cut of this part of the membrane to form a vesicle which is released inside the cell in the cytoplasm. So this is now called a clathrin-coated vesicle. Clathrin is then rapidly uncoated, and this uncoated vesicle then fuses with the early endosome, which is Rab5 positive. From here, as I mentioned earlier, the fate of the molecule can either be, because it is recycled, back to the membrane, or it is targeted for degradation. Please feel free to stop me if there are any questions in the middle. Okay, so clathrin, as I mentioned, clathrin, as I mentioned, it's basically it's a building block of endocytic vesicles. There are two types of clathrin. There are two genes which encode for two different types of clathrin molecules in the cell. There are the heavy chains and the light chains. So the heavy chain is what is shown in this teal or greenish color, and the light. Together they assemble to form this three-legged structure called a triskelion. This triskelion then interacts with many more triskelions to form a cage-like structure or a basket-like structure, which, as you can imagine, provides rigidity and shape to the part of the membrane which needs to be internalized. So what we found was that knocking down the clathrin heavy chain basically resulted in a decrease in embryonic stem cell pluripotency. How did we assess this? So on the left, you can see, the wild-type embryonic cells, which are stained with alkaline phosphatase, and you can see this lovely red staining and these nice, the morphology of colonies. When you remove the clathrin heavy chain, you observe two things. One is that the cells no longer form colonies, and secondly, the amount of alkaline phosphatase staining has dramatically reduced. We also got the same results when we used a dominant negative form of dynamin, which is also an integral part of clathrin-mediated endocytosis. Together with this, we saw a decrease in the expression of stem cell markers such as OCT4, SOX2, NANOG, and KLF4. So the dotted line here represents the expression of these markers in wild-type stem cells. The moment we remove clathrin heavy chain, which is shown in black, or express a dominant negative form of dynamin, which is shown in light gray, you can appreciate that the level of expression of these markers reduces considerably compared to the wild-type cells. At the same time, there is an increase in the expression of differentiation markers, which mark the different cell lineages. And again, here at 1 are your wild-type cells, and you can appreciate that when we knock down clathrin, the levels of all these markers increases, suggesting that the cells are no longer now behaving like stem cells, but are moving closer towards differentiated cells. The other thing that we noticed was that knocking down the clathrin heavy chain caused a loss of the epithelial nature of embryonic stem cells. So mouse embryonic stem cells are cells which do, have certain properties of epithelial cells. They like to stick together. They like to stay together. And we found that when we knock down the clathrin heavy chain, these cells now disperse. They no longer attach to one another. And this was accompanied by a loss in the expression of the cell-cell adhesion molecule E-cadherin or ECAD. So in wild-type cells, this Western blot shows you that there is enough amount of expression of E-cadherin. That expression goes down when you knock down the clathrin heavy chain. The expression of other epithelial markers, such as CRUMS3, epithelial cell adhesion molecule and occludin also go down. And the expression of mesenchymal markers, such as N-cadherin, ZEB1, snail, and slug also increase, suggesting that these cells are now moving away from an epithelial state and are moving closer to a mesenchymal state. So I mentioned E-cadherin. What exactly is this? This is a calcium-dependent cell-cell adhesion molecule. It's a transmembrane protein, and it forms these, interactions, these trans interactions between E-cadherin present on two different cells with the help of calcium. So these molecules help to hold cells together and form cell-cell adhesions. So we then asked, because we saw this phenotype of the cells dispersing and of a decrease in E-cadherin, whether there was any connection between clathrin-mediated endocytosis and E-cadherin. And what we noticed was that E-cadherin is internalized through clathrin-mediated endocytosis. We saw this because we saw colocalization between E-cadherin and clathrin heavy chain. So these are immunocytochemistry images which show you the localization of these molecules in stem cells. And the zoomed in picture here on the right can show you that there is abundant colocalization between these two molecules. We also found that E-cadherin is recycled back to the membrane in a Rab11 dependent manner. So Rab11 is a marker of the recycling endosome. And once again, when we stained embryonic stem cells for Rab11 or E-cadherin, we found that there were significant regions within these cells where there was a colocalization, indicating that E-cadherin is internalized only to be recycled back in a Rab11 dependent manner. We then blocked recycling by using a drug called bafilomycin A1, and we noticed that under these conditions, two things happened. One is that the relative expression of the pluripotency markers, such as OCT4, SOX2, NANOG, and KLF4, decreased compared to the vehicle control. And secondly, that when we treated cells with bafilomycin, there was an accumulation, as you can see here, here, and here, of E-cadherin in and then this results in a downstream signaling which turns on a lot of mesenchymal genes. So we then asked whether TGF-beta expression also changed and whether there was any relationship with the clathrin-mediated endocytosis. So we noticed that a TGF-beta, when we stained cells for the TGF-beta receptor and clathrin heavy chain, again these colocalized in vesicles, suggesting that TGF-beta receptor was maybe being internalized through clathrin-mediated endocytosis. We also found that when we knocked down the clathrin heavy chain using siRNA, there was an increase in the amount of TGF-beta receptor 1 protein in these cells and also in the downstream signaling. So here you can see the increase in the phosphorylated form of SMAD2 and 3 in cells which lack clathrin heavy chain. What we also knew was that this molecule is targeted for lysosomal degradation. So this is a picture I showed you before where a TGF-beta receptor colocalizes with the clathrin heavy chain. We looked then for which compartments within the cell this molecule is present. And we found that it colocalizes with the late endosomal marker Rab7, which you will appreciate here in the zoomed up image, that you can see that the red and the green colocalize to make these very nice yellow puncta, indicating that the TGF-beta receptor is normally present in the late endosome in stem cells. So we then asked what happened to TGF-beta receptor levels when we knocked down the late endosomal marker RAB7. Again, we noticed an accumulation of the TGF-beta receptor and an increase in the downstream signaling, which is denoted by phosphorylated SMAD2 and 3. So then we asked, this loss of pluripotency when we knock down a CME, it seems to be happening because of two things. One is because E-cadherin levels are going down, and the second is because TGF-beta levels are increasing. So can we rescue this by blocking TGF-beta signaling? And we did that using these two drugs, Repsox and SP543-2, I think. Sorry, that number eludes me right now. But what you can see here on the panel in the left is when we stain cells for alkaline phosphatase, the expression of alkaline phosphatase and the ability to form colonies is rescued in both the cases when you block signaling downstream of TGF-beta, even in the presence of the siRNA against the clathrin heavy chain. And this is a quantitation of colonies which are now OCT4 positive and E-cadherin positive, again showing that there is a rescue in the number of colonies both here as well as here compared to here where you have just the CLTC knockdown. So what we want to say is that clathrin-mediated endocytosis works by balancing opposing signals. In wild-type cells, you have E-cadherin, which gets internalized and then gets recycled in a RAB11-dependent manner. This maintains embryonic stem cell pluripotency. In these same cells, the TGF-beta receptor 1 gets internalized again through clathrin-coated vesicles, but is then targeted for lysosomal degradation through the RAB7 pathway. And this together maintains embryonic stem cell pluripotency. Now, when you knock down the clathrin heavy chain, what happens is that the TGF-beta receptors are no longer internalized and they are not targeted for degradation. This then strengthens the signaling downstream, results in an increase in phosphorylated forms of SMAD2 and 3, and this drives the expression of a mesenchymal cascade, which then ultimately shuts off epithelial markers such as E-cadherin. And then this then converts the cell from going from a stem cell to more of a differentiated cell. So this work was published last year in Stem Cell Reports and was done almost single-handedly by a graduate student in the lab, Narayan. So to sort of give you an idea, we believe that stem cells seem to have some sort of an endocytic threshold, and this is regulated by levels of clathrin-mediated endocytosis. If you decrease the level of clathrin-mediated endocytosis to a certain level, you can then force an embryonic stem cell to differentiate. We are now examining what happens in the reverse situation, where you can take a differentiated cell and push it towards an embryonic stem cell through the process of reprogramming. I'm going to just switch and just talk about for another 5 minutes about a different story that we're doing. So like I mentioned, there are two types of clathrin molecules. There are the heavy chains, which I spoke about just now. And then these also associate with smaller molecules, which are called the light chains. And the light chains play a very important role in regulating the type of coat that is made. Okay. So what is known in mammals is that the clathrin triskelion is made up of 3 molecules of heavy chains of clathrin and 3 molecules of light chains. Polymerization of the clathrin triskelions form the clathrin coat. Now in mice, a single gene encodes for the heavy chain, whereas there are 2 genes, CLT-A and B, which encode for the light chains in mice. However, the roles of these light chains is so far unknown. So we decided to ask whether we could use gene editing techniques to investigate the individual roles of these 2 genes in early mammalian development. So just a bit of introduction about these. So the light chains A and B, they arose due to local duplications around 600 million years ago, and they share around 60% identity. Both light chains share a conserved stretch at the end terminus through which they can bind certain actin-binding proteins. And there are a large number of splice variants of the light chains, which are mostly present in the nervous system. So as I mentioned, we decided to use gene editing techniques through which you can specifically cut or modify a part of the DNA. Specifically, we use the CRISPR-Cas9 system, which gained recognition last week because the Nobel Prize in Chemistry was awarded to these 2 women, Emmanuelle Charpentier and Jennifer Doudna, for developing a method by which genes could be edited in almost any cell type. So how this technique works is through the help of a guide RNA, which is in maroon here, which binds to this green protein called Cas9. The guide RNA provides sequence complementarity and can then detect a specific sequence in the genome, which can then be cut with the help of this protein. So using the same technique, we decided to build knockout embryonic stem cells and mice for the clathrin light chains. So the details don't matter, but we used guide RNAs and Cas9 to target certain specific exons within the gene so that we could try and get a knockout. So we have successfully built knockouts for these genes in embryonic stem cells. We also managed to build a truncation mutation for one of these genes in mice. And the way we did this is, so if you think back to one of the first slides that I showed, the fertilized egg has, if you remember that video, which looked something like this, it has these two pronuclei. This is right before the two nuclei fuse together. Okay, this is soon after fertilization. So you have the male and the female pronucleus, and you can easily detect this under the microscope. So using an extremely fine needle, what one can do is one can deliver, your mixture of a guide RNA as well as the Cas9 protein, or you can use RNA, or you can use DNA, and you can inject it into this prominent pronucleus. And then you can allow the zygote to develop and transfer this to the oviduct of a pseudo-pregnant female to allow implantation and then production of a transgenic mice. So we injected a number of embryos and we transferred a number of embryos and we established some founder lines. We were trying to make a knockout of a light chain, but instead what we got was a truncation. And we managed to make these mice, which basically express a truncated form of one of the light chains. And this was built by CRISPR-Cas9 genome editing, as I said. So what we built was this. So this, the first line in purple is your full-length protein for the clathrin light chain B. What we ended up building was a truncation which expressed only the first 84 amino acids of the clathrin light chain B. And so these mice were expressing a mutant form of the protein. This was the phenotype of these mice. These mice initially displayed a very severe mortality. We had two founder lines. We've just called them type 1 and type 2. And we found that both of them, if you look at this Kaplan-Meier survival curve, you can see that compared to the wild type, which is shown in black, the blue and the red lines are the two different founder lines. These displayed significant mortality around 40 to 60 days, and pretty much all the mice died. And they seemed to die because of some sort of an inflammatory response where, they had, very severe diarrhea, which was mixed with blood several days prior to death. And they also had a very inflamed rectum. So these mice also displayed severe intestinal abnormalities. So here is the top panel is basically wild-type mice, and the bottom panel is mutant mice. And what you can see is a complete destruction of the small intestine and the large intestine in these mutants, and a huge infiltration of immune cells in these mice. Difficult to characterize, and I just wanted to, bring this up to show you that one can use a combination of, looking at early embryos as well as using gene editing techniques to try and understand what genes do during development. So this is my last slide. So I just wanted to show you that we can build these mutant mice. We've seen they display severe mortality. This mortality is accompanied by intestinal defects, diarrhea, hinting at defects in the intestinal epithelial development. And we're trying to understand the cause of these defects in these mice. So, of course, all of this work was done by a very talented group of people in the lab, and also with help of very generous funding from various sources and support from NCCS. And thank you very much. I'd be very happy to take questions now.
Thank you very much, ma'am. It's a very, very excellent talk, and I must congratulate you for working this kind of work at your institute and your lab. We are very much fascinated now. The stem cell research future is very, very nice. You can see here what NCCS is doing, and you people are doing very nice work at there. So now I just request to Dr. Nivedita Lanka ma'am for conducting a discussion, and also I request Arpit to take few queries for the speaker so that it can be resolved. So now session is open, and this session is open for question and answer, and also for discussion. Please.
Thank you so much, sir.
Okay.
Yeah,
So
please
all the
go ahead.
presentations.
Arpit, firstly, Dr. Nivedita ma'am will conclude, and we will start.
Okay,
Yes, ma'am,
thank
please.
you.
Okay, thank you, Dr. Chaudhary. So today morning we had 3 talks. Unfortunately, the second one, I think I had some connection problem. So after the first or second slide, I could not see or hear anything. But what I could, and I could actually start with her talk only, Dr. Parul Jori. She was discussing what I understood overall, like she was discussing about the bioinformatics aspects, how one can utilize the knowledge of bioinformatics in addressing various issues in stem cells, like the data sets that we get. So by different techniques like clustering and enlistment analysis, something called mutational calling. I am not that good at bioinformatics, but that's what I was interested in actually listening to her talk. Unfortunately, I could not. So I think all of you must have enjoyed her talk thoroughly. So this is actually the one of the tools where one can utilize the mathematical modeling and AI-based approach in addressing many aspects. So whatever you do on bench, and to understand what are the aspects like how temporospatially different gene regulation is taking place, which one are the ones which are undergoing mutation, or which one the ones we have to actually focus more on. That's what we require some tools, the bioinformatics in silico tools, after which we can actually validate those. So that way actually her talk must be quite inspiring, I guess, which I could not unfortunately attend. And then Dr. Deepa is my colleague here at NCCS doing wonderful work. And as she deliberated actually the importance of endocytosis. So as we know, this is a process actually for the trafficking, cellular trafficking, and this is one of the important factors also. Because when cell-cell communication is taking place, this is also one of the important aspects that is what is involved. And she has shown very nicely on her presentation. Lucidly, she has demonstrated how important endocytosis is for maintaining the pluripotent nature of embryonic stem cells. And there also she has very nicely showed Catherine-mediated endocytosis in particular. That is what is involved. And the live chain, she has also done the editing technology, gene editing. By that, even most interesting part is they have developed the mutant mouse now, where they are studying further. And during her talk, she has also showed how E-cadherin and the clathrin mediated, how the inverse correlation is there. E-cadherin is going down and TGA-beta receptor is going up. So through that, she has demonstrated the signaling cascade which are actually responsible during this process. And very nicely, she has demonstrated all this. And I think these also must have brought a lot of curiosity among the young researchers and the students alike. And coming to my talk, it was a much broader aspect, the general overview of what the stem cells are, different types of stem cells, the categorizations, how one can get stem cells, and how one can characterize. I did not go much to the characterization part, but at least how one can differentiate and see the pluripotency or multipotent nature of stem cells depending on the source of their derivation, and the implications, various implications, right? Like as a basic biologist, as a researcher, what one can do in the lab, further what can have the industrial implications and in clinics. And the most recent advancement pertaining to tissue engineering, that is what actually I focused on. I think all of you must be having a lot of queries, and so please feel free to ask us. So whatever possible way we can address, all 3 of us, we will do that. Our best. Thank you all. So can we go for the question answer session now?
Yes, ma'am. So thank you for your wonderful words. And now we have some questions and queries from our participants. And initially, we are going to start with Nivedita ma'am. The first question is for you only. And the question is from Priyanka Da Costa. She's asking why it is difficult to cultivate muscle, fat, and other components together.
Difficult to cultivate muscle, fat,
And other components together.
if I understood your question correctly, you mean to say that whether you can co-culture muscle cells and fat cells and other component. I don't know what you exactly mean. So if you want to do co-culture, I don't think it is difficult, because if you know mesenchymal stem cells, those have the propensity or the intrinsic characteristics of giving rise to bone, fat and cartilage. So they are, if you want to direct the cells to differentiate into particular lineage, you can provide specific factors or the cocktails, then you can direct those. Embryonic stem cells, they have got the pluripotent nature. So when you are doing spontaneous differentiation, what Dr. Deepa was showing also, during spontaneous differentiation, you in fact see all the three germ layers. So, but directed differentiation, you get majority of the cells going to different cell type, like a specific cell type of your interest. But if you want to co-culture, you can take the cells, and there are different ways of co-culturing cells. So there are also, it's called, I'm just blanking. So you can grow two cell types, one at the bottom, and the migration assay where we do Bowden's chamber. So there also you can culture the cells, or in the same dish you can culture two cells to see the cellular interaction. So if you label one cell type with one fluorescent marker, another cell type with another fluorescent marker, you can see also the connectivity between those and how they communicate with each other. Yeah. Is it okay? Like, could you get my point, or there is any further query?
I guess the participant had got your point. So thank you so much for the answer. The next question we have for you from Dr. Vaishali, and she's asking, can we use stem cell engineering for the blood cells? Is there any research has been undertaken for the same?
Okay. Stem cell engineering for blood cells. Yes, engineering if you are talking about, so they are, if you want to produce more cells, so for the cell replacement, yes, it is possible. You can take directly from the, if it is autologous way you want to do the therapy, obviously you can take from bone marrow, and that is the usual way people are trying also, and that has gone to clinic. But if you want to generate blood cell types, specific cell types, that you can take adult stem cells, or if you want to take iPSCs, one can direct the cells to a specific, to the hematopoietic lineage, and you can also generate the cells of interest. But engineering, we are talking about 3D, because blood cells, those don't have a 3D structure per se. So in that case, most of the 3D organs you can engineer, but you can get the cells of your interest by doing the cultural manipulation in vitro. In a 2D culture itself, you can do.
So thank you so much, ma'am.
Our institute itself, one of our former colleagues, Dr. Kale, he was in fact trying to do from the adult bone marrow derived hematopoietic stem cells, and there they have shown also specific factors of artificial blood you can also make. Such kind of research also is going around.
Thank you so much, ma'am, for providing such a big information on this. And now the question is for Dr. Parul Johri, ma'am. Dr. Parul Johri, ma'am, if you are present here, just please open your mic. Dr. Parul, ma'am, I'm requesting you to please open your mic so that we can interact with you with some queries.
Please unmute, Dr. Parul.
Can we go with Arpit? Dr. Deepak?
Yeah, you can go to the next question. Maybe Dr. Parul is not around.
Now she's available. Yeah. Arpit, you may ask the question. Arpit?
Yes, so Dr. Parul Johri, ma'am, are you there?
Yes.
Okay.
Yes, I'm there. Can you listen my voice now?
Yeah, you are able to.
Okay. Fine. Thank you. Yes, Arpit, can I have the question?
Yes, ma'am. So the first question for you is, could you please explain the principal component analysis?
Okay. Principal component analysis is basically a dimensionality reduction technique. So dimensionality reduction means if we have data in more than 3 dimensions, we need to reduce it to 3 or 2 dimensions so that we can plot a graph and we as humans can analyze the data and can interpret it. So principal component analysis is basically dimensionality reduction. We are reducing the dimensions in which our data is being scattered. Like if I'm comparing a set of, if I'm in a class, there are 50 students and I'm comparing their variations in their skin color, eye color, length of their hair, their height and their weight. So out of these 5 parameters, I have reduced them to only height and weight, which will have the maximum variation. So we are basically looking into the variables which are having the maximum variation, and we take those variables and we analyze the data taking only those variables. So that is called as dimensionality reduction in PCA.
Thank you so much, ma'am, for your answer. The next question we have, could you please elaborate the lineage biasness problem with respect to computational tools?
The lineage biasness means we are trying to find out the lineage hierarchy. Lineage means our ancestors, their own ancestors, from where we have descended. So in genes also, like initially there was a single gene, then some mutation has happened and the gene has changed, and again some mutation has happened and the gene has again changed. So the evolution of the gene from one section to the another, so that is called as the lineage biasness. So certain changes are biased to certain genes, so that biasness we are checking in this particular section. Like in codon also, you would have heard about codon biasness. So certain codons are biased to prokaryotes and certain codons are biased to eukaryotes. So we have a lineage biasness also, and that parameter is being thoroughly identified using bioinformatics algorithm and tools. Recently they are developing for that.
So thank you so much, ma'am. That's all from the questions for you. And now I'm inviting Dr. Deepa Subramaniam, ma'am, for the answers of the questions we have received for her. So, Dr. Deepa, ma'am, we have a question for you.
Yeah.
Yes, ma'am. So the first question is for you, that is, how many days are used for culturing a stem cell?
So it depends on what exactly, that means. One way to interpret it is how long does it take to establish a culture of stem cells. If you start from an embryo, that could take a few weeks to initially establish the culture. Once the culture is set, you usually, in the case of mouse embryonic stem cells, you would split the cells every 3 days or so. In the case of human embryonic stem cells, you would passage them or split them. Hello, can you hear me?
Yes, ma'am, you can continue.
Okay. So, no, it told me I was logged out. So, in the case of human embryonic stem cells, you would do this once every 5 days or so. So, it depends on the type of stem cells. It depends on how long it takes to establish the culture. So, there really isn't a fixed answer for this. It depends on which type of stem cells you're culturing.
Thank you so much, ma'am. The next question we have for you, what is knockout gene technology?
So knockout gene technology is basically, using some genetic method to remove or to knock out a gene, to remove a gene and to understand the effect that the removal of that gene would have on a cell or an organism or a tissue or whatever it is you're studying. Now, you can remove the gene or you can knock out the gene, using a number of techniques. The one that I mentioned right now today was, CRISPR-Cas9 gene editing. Before that, there were many other, techniques which could be used to remove a gene. So whatever is the technique that one uses, the idea of knockout gene technology is to knock out or remove a gene to understand its effect and its function in a particular cell type or a process or an organism.
Thank you so much, ma'am. The next question we have, is there any cell line that do not express clathrin?
That's a great question. So it depends on which type of clathrin you're talking about. The heavy chains, we believe, are present in almost every single cell type. They are present at different levels. We do not yet know if a cell or an organism would be able to tolerate if there was absolutely no clathrin. So we have used clathrin knock down, not knock out. So that's something to keep in mind. The light chains have a tissue-specific distribution, and some light chains are preferentially expressed in only some cell types. For example, the light chain A is expressed only in the B cells of the immune system. So there are differences in that case, but we do not yet know whether certain cell types can tolerate a complete loss of the clathrin heavy chain.
Thank you so much, ma'am. The last question we have from Priyanka. She is asking, how can this be used in food sector and what are the setbacks faced in the same?
This question is to whom?
This is just, I received this question, so yes, sir.
Actually,
Okay, can I answer that?
Yeah.
Can I?
Yes,
Okay.
yes, ma'am, definitely.
So she must be actually talking about the tissue engineering part, the food sector. So I think last year maybe there are a lot of news in this regard that's coming. So whether you can engineer meat, those who are non-vegetarian. So instead of using the animal side, whether you can engineer, because meat is basically the muscles. So you can actually engineer muscle cells and which can substitute for the animal meat, but whether the taste will be similar to the animal derived ones, that is questionable. But that is not actually the food sector application from the animal side. If you are thinking, this is what the meat substitute people are thinking of engineering. But from plant side, of course, we all know plants anyway, that is our basic food, and vegetarian especially like tea and others. So definitely the engineering, that though already we know the plants are totipotent cells, plant stem cells are totipotent, and we are getting. So the engineering aspect there, we already know how they do also by hybridization and different varieties production. That is different, but animal tissue engineering aspect, this is mostly it is done for the meat production so far.
So thank you so much, ma'am. Over to you, Dr. Kamlesh. Sir, that's all from the QA session. Thank you so much.
Thank you very much, everyone. And first of all, I would like to thank Dr. Nivedita Lanka, ma'am, for encouraging us and supporting us and accepting our invitation to be as a convener of this particular workshop. And she has conducted well this program. And I also thankful to Dr. Deepa Subramaniam, ma'am, and she has taken some pain because her lecture is late. And to be present here and given so much very nice information regarding this particular stem cell research. And I also would like to thank Dr. Parul ma'am to be here. So before leaving this session, I would like to invite Arpit for giving formal vote of thanks to all these speakers. Yes, Arpit.
Thank you so much, sir, for giving me this opportunity. So today's stories come to an end. The more we go deeper in ocean, we reveal a new life. Similarly, with stem cells, more explorer has to understand the origin of life. With these words, we, the team of Biotech e-Refresher course, would like to thank Dr. Nivedita Lenka ma'am, Dr. Sugandha Singh ma'am, Dr. Parul Johri ma'am, and Dr. Deepa Subramaniam ma'am, presented as women cutting edge in recent advances in stem cell engineering. Thank you so much. I would like to thank our participants for joining with us. I would also like to thank Dr. Kamlesh Chaure for his wonderful efforts in organizing such an intellectual workshops. And I'm also giving sincere thanks to Vivek Agnihotri for broadcasting the entire session. I would also like to thank the entire IT team of AKS University and the Management Authority for making this e-refresher course a top on the sky. So thank you so much, every presenters and the organizers.
Thank you, Arpit, and
Thank you very
once.
much.
Thank you very much, ma'am.
Thank you, Dr. Chaudhary and everyone. Thank you.
Yes, ma'am. I will invite you in future, and we will try to learn more things because our students are also visiting NCCS regularly for their one-year project work. Our 3-4 students has conducted research there. So we are very much fascinated with NCCS and activities, research activities going on there. And I always see the updates on the website that what NCCS is doing right now. Thank you very much, ma'am. And I would like to let you know that in this whole e-refresher course, which is just we are conducting since 3rd of September and which is going to be finished on 21st October. And most of the speakers belong to the, we can say that Nari Shakti. So thank you very much once again. Now we are stopping the broadcasting of this event.